Hello, everyone. My name is Diana Wall, and I am the director of the School of Global Environmental Sustainability at Colorado State University, and also the chairperson of this National Academies Study Committee. In my role as chair of this study and as moderator of this committee meeting, I'd like to welcome everybody here in the room in Washington, D.C. It's nice to see everybody in person. And those of you watching the live stream and all of you who may be watching this later. The task being undertaken by this committee of the National Academies of Science, Engineering, and Medicine is to explore the linkages between soil health and human health. The committee's full statement of tasks can be found on the study website, which is available at the link under project to the right of the embedded video if you're watching this online. I'd like to give you some sense of the format of today's meeting. I will shortly turn to the committee members and ask each of them to unmute and introduce themselves. You can also find a short biography of each member of the committee on our study website. I will then turn things over to Karolini, the study director with the National Academies, to provide a brief overview of the study process. Following her presentation, I'll introduce our speakers for today, and we will adjourn the meeting at 5 p.m. Eastern Daylight Time. I want to note that this is an open, on-the-record session. Interested individuals and members of the press have been invited to join us as observers. This is not a news conference, however, and we will not be entertaining questions from the audience. Reporters who would like to talk with members of the committee are kindly asked to touch base with the staff officer, Karolini, and also anybody who is interested in submitting a written comment or a document to the committee is welcome to do so by clicking the Provide Feedback link on the right side of the study website. You can also subscribe for updates about the study by signing up at the National Academies Board on Agriculture and Natural Resources listserv, also on the right side of the page. I'd like to remind everyone that this is an information gathering session. That is, the committee is in the process of assembling materials that it will examine and discuss in the course of making its findings, conclusions, and recommendations. Therefore, I ask everyone to please be extremely mindful of the fact that the committee has made no conclusions yet, and that it would be a mistake for anyone to leave the meeting thinking otherwise. Comments made by individuals, including members of this committee, should not be interpreted as positions of the National Academies, of the individuals, or the members of the committees. In addition, committee members typically ask probing questions in these information gathering sessions that may not be indicative of their personal views. The committee will deliberate thoroughly before writing its draft report. Moreover, once the draft report is written, it must go through a rigorous review by experts who are anonymous to the committee, and the committee must then respond to this review with appropriate revisions that adequately satisfy the Academy's Report Review Committee and the National Academies of Sciences president before it is considered an official Academy report. At this time, I'd like to ask each committee member, when I call your name, to turn on your microphone and briefly share your area of expertise and your current position. Sam, we're going to start with you. Sam Banerjee.
Hi, everyone. My name is Sam Banerjee. I'm from the North Dakota State University, and I'm in the microbiology department there. So in my lab, we study microbiome science and the factors that shape microbiomes in soil. And we try to understand the molecular dialogue that happens between plants, soil, and microbes. So thanks.
Nick Pasta.
Hi, I'm Nick Pasta, Ohio State. I'm a professor of soil and environmental chemistry. Most of my work is looking at contaminant chemistry, bioavailability, and remediation.
Sarah Collier.
Hi, I'm Sarah Collier, an assistant professor in food systems, nutrition, and health at the University of Washington in Seattle. And my background is in crop and soil sciences and My work focuses on food and agricultural systems sustainability and resilience.
Carlotta Dale.
Hello, my name is Maria Carlota Dao. I'm an assistant professor at the University of New Hampshire, and my areas of research include nutrition, the gut microbiome, cardiometabolic health, and health disparities.
Mike Grusak, he's on Zoom.
Good afternoon. This is Mike Grusak with the U.S. Department of Agriculture, Agricultural Research Service. My background is in plant physiology, but also human nutrition, and the link between nutritious foods and human health.
Thank you. Colleen? Neil?
Good afternoon. My name is Callie Neal. I'm a professor in the Department of Animal and Food Sciences at University of Delaware, where I work in microbial food safety, in particular looking at survival and transfer of viruses, protozoa, and bacteria onto crops related to fresh produce.
Rebecca Nelson.
Hi, I'm Rebecca Nelson, a professor at Cornell University. I'm on the faculties of plant science and global development. And after a couple of decades of working as my lab working on plant genetics, disease resistance, I've been dedicating myself to the idea of the circular bionutrient economy. Thank you.
Kate Scow.
I'm a professor in Department of Land, Air, and Water Resources, soil microbial ecologist at University of California, Davis, recently emerita, and worked a lot on bioremediation of organic contaminants in soil and groundwater, and also the microbial underpinnings role of microbial diversity in agroecosystems.
Thank you. Anne Skillis-Ray.
I am Anne Skillis-Ray. I'm an assistant professor at the University of Arizona in the School of Nutritional Sciences and Wellness, and my research is about interventions to improve human health through cardiovascular risk factors using diet and dietary supplements.
Kelly Wright.
Hi, my name is Kelly Wrighton. I'm a professor in the Department of Soil and Crop Sciences, and I'm co-director of the Colorado State Microbiome Network.
Ilva.
Hi, I'm Eva Lexberg. I am with MPG Ranch at University of Montana. I'm a soil ecologist, primarily focusing on agricultural management practices and soil ecosystems.
Lindsay Slaughter.
Hi, I'm Lindsay Slaughter. I'm an associate professor of soil microbial ecology at Texas Tech University, and my work focuses on soil health and microbial communities in agricultural ecosystems and stressful environmental conditions.
Thank you. Have I missed anybody that's here in DC? Okay. Katrina Abubara and Joseph Owika are not here. You can find all the committee bios on our website. I'd now like to turn things over to Carol Laney, who is the Senior Program Officer with the Board on Agriculture and Natural Resources at the National Academies and the Study Director of this activity. She will give us a brief overview of the National Academy study process. Kara?
Thank you, Diana. I just want to say a few words about the National Academies for anyone joining us today in the room or online who might not be familiar with the institution or our processes. The National Academies was founded in 1863 by in a bill passed by Congress and signed into law by President Lincoln. Later, the Academies was also granted the authority to create the National Academy of Engineering and the National Academy of Medicine, which was previously known as the Institute of Medicine. So today we're the National Academies of Sciences, Engineering, and Medicine, and we consist of the 3 honorific bodies, but also the work of scientists across the country, inside and outside the Academy's societies. And these experts provide advice to the nation involving all manners of questions related to science, engineering, and medicine. The mission detailed in the Congressional Charter is to investigate, examine, experiment, and report upon any subject of science or art whenever called upon. And Kat, if you go next slide, thank you. We've fulfilled this mission through many convening activities, including consensus studies like the one that we're convened for today. Just to share a little bit more detail about consensus studies as opposed to other activities that we do, such as forums or workshops. With a consensus study, we have a committee that's convened specifically for a unique statement of task. Experts who serve on these committees do so as individuals, not as members of any institution or organization to which they may belong, and they also serve as volunteers. Consensus study committees review the state of the scientific literature in order to write a report that responds to their unique statement of task, and that report will hopefully be used to inform sound policies and public opinion and advance the pursuit of science in the specific field in question. Each consensus study goes through a number of phases. It starts with the study being defined. That's a negotiation of the statement of task between the study sponsor and the National Academies. The sponsor for this particular study is the U.S. Department of Agriculture's National Institute of Food and Agriculture. Once the statement of task has been agreed upon, the National Academies seeks that will represent not only the expertise that is needed to complete the study, but also the relevant points of view. Nominees are appointed to the committee by the president of the National Academy of Sciences, Dr. Marcia McNutt, and committee membership is provisional until a discussion of conflict of interest, disclosure, and composition has been held by the committee at its first meeting. So then we move into the informational gathering stage, which is the point of the study process, which we're in now. Today is our last hybrid meeting that we have planned that has an information gathering component to it. In meetings like the one today and the several that are recorded online that you can look at if you'd like, the committee gathers information relevant to its statement of task. We're also moving into the deliberative stage of the study where we discuss what needs to be in the draft, what we can come to consensus on, what we can't, and we begin writing. And once the draft report is complete, their draft is submitted to a group of reviewers, as Diana said. The reviewers submit comments that the committee must respond to and revise the report accordingly. And when the National Academies Report Review Committee deems that the committee has appropriately responded to reviewers' comments, then the report is approved for public release and delivered to the sponsor, as well as made publicly available. I just want to say a couple words about open versus closed sessions. So our information gathering sessions are held in open session, which means that not only is the committee and the staff present, as well as our invited speakers, but anybody may be present. And these meetings are posted online in advance and they are streamed and recorded. When the committee meets in closed session, it's just the committee and staff. And we meet in closed session so that the committee has the opportunity to deliberate on the statement of task and to change their minds to come to conclusions without undue influence from outside individuals or opinions. This is just briefly the study timeline. So as I mentioned, we're wrapping up our information gathering phase. We're moving into our heavy report writing phase, and we hope to submit the report for review by the end of the year and to have it released to the public in the spring of 2024. Let's see. Here's our statement of tasks. If Kat goes to the next slide, we have a lengthy statement of tasks, so I'll just give everyone a moment to read it. And Kat, you can go to the next slide. As I said, USDA's National Institute of Food and Agriculture is the sponsor for this study, and it was originally came to USDA in a request from in congressional language from the last appropriations bill. Finally, this is a screenshot of our study's website. You can see on the left where you can hear about where you can find our past events, as well as the statement of task and the committee bios. And on the right in the oval, you see the link where you can provide feedback for the committee. That includes any documentation you would like the committee that you think would be pertinent to the committee to look at for its statement of task, as well as to subscribe for updates about the status of the study if we have future meetings when the report will be coming out, those sorts of informational items. And my email address is here if you need to reach me, if you have any questions about the study, as well as my colleague, Kat Kane. And with that, I will turn it back to you.
Thank you. Our plan today is to hear from 4 speakers and to conclude the open session at the 5:00 p.m. Eastern time with a short break at 3 p.m. It is my pleasure to introduce our first speaker today, Dr. Jo Handelsman. She is the director of the Wisconsin Institute for Discovery and a Villas research professor and Howard Hughes Medical Institute professor in the Department of Plant Pathology at the University of Wisconsin-Madison. She joined the faculty at UW-Madison in 1985, where she served for 25 years before moving to Yale University in 2010. From 2014 to 2017, she served as science advisor to President Barack Obama in her role as Associate Director for Science in the White House Office of Science and Technology Policy. After leaving the White House, Jo returned to University of Wisconsin-Madison, where she continues to work on national policy as well as direct the Institute and pursue her own research on the soil and human microbiomes. In 2021, she published the book, A World Without Soil, a book that presents the soil erosion crisis and policy recommendations to avert it. Jo is known for pioneering the field of functional metagenomics. She has been elected to the National Academy of Sciences, the American Academy of Arts and Sciences, and the National Academy of Engineering. National Academy of Inventors. Received the Presidential Award. Sorry, I didn't have my microphone on. Then the Presidential Award for Excellence in Science, Mathematics, and Engineering Mentoring from President Obama in 2011. Thank you for joining us today, Jo. It's wonderful to have you.
Sorry, I had trouble getting unmuted. Thanks for having me. And do my slides appear okay to everybody? You can see them?
Yes, they look great.
Okay, great. Well, thanks for having me. And it's a real honor to contribute to the deliberations of this committee, which I think is a really important one. And I'm just thrilled that the Academies has taken on this topic of the linkages between soil health and human health. Today, I'm going to talk very briefly about a program called Tiny Earth and the impetus for the program, which is antibiotic discovery. So I'll start with the antibiotic crisis, just to make sure everybody knows the severity of the situation. I'll talk about Tiny Earth, and I'll describe the pipeline we've developed to optimize discovery of antibiotics. We probably all know that Alexander Fleming discovered penicillin in 1929. But it wasn't until the 1940s, during World War II, that the work was done to optimize production of penicillin to create enough of the drug to ship over to the troops in Europe. And so it was a collaboration of Ken Raper's group at the USDA, his technician, now known as Moldy Mary, but her name was actually Mary Hunt, and Elizabeth McCoy, a professor at the University of Wisconsin, who together isolated high-producing strains and then mutants of those strains that produced vast amounts of penicillin. From the 1940s to the 1980s, the pharmaceutical industry took antibiotic discovery very seriously. They discovered many antibiotics, literally hundreds during that period of time, and most of them were from soil. But because of the overuse, abuse, and misuse of antibiotics, both in human and animal agriculture, human health and agriculture, we immediately, as soon as many of these antibiotics had been released, started seeing the rise of resistance. And here you see three of the major pathogens that are becoming resistant, with the most serious one being MRSA, which is methicillin resistant, Staph aureus, which is a very serious, particularly hospital-borne disease that is often incurable these days because the antibiotics that once managed it are no longer working. Simultaneously, in the 1980s, when resistance was starting to be recognized as a serious issue, the industry abandoned antibiotic discovery. And they said that this was because the soil was tapped out. There was nothing left to discover. But in fact, if you get inside most of these companies, you find out that it was entirely based on bottom line decisions. Antibiotics are not very lucrative. People take them for a few days or at maximum a few weeks in most cases, and the companies turn their attention to chronic disease for drugs that are taken for a lifetime. And so here you see on the right, the drop in antibiotic discovery and release. These are applications for new antibacterial drugs. And you can see that it started at a pretty high clip. We were releasing many antibiotics in every 5-year period. It dropped down in the 2000s to as low as 3 in a 5-year period. And it has started to creep up, but I understand this is starting to level out. So we now have a crisis with many pathogens that were once treatable with antibiotics being untreatable. There are 3 million antibiotic-resistant infections just in this country. Worldwide, of course, it's many more. And the CDC predicts that antibiotic-resistant bacteria will be the leading cause of death by 2050. So the combination of the national crisis and my own mother's death from antibiotic-resistant bacteria has fueled my career in discovering antibiotics from soil organisms. That quest started, in fact, as an accident. We just started stumbling upon antibiotics, and then as the industry pulled out, my lab was discovering more and more antibiotics from soil organisms. And I knew that this myth just couldn't possibly be right, that the soil was in fact not tapped out. There's still a large market for antibiotics. So even though they may not be the most lucrative drugs on the market, we're still talking $40 billion a year in sales, and that's going up all the time. Most of this, of course, is just rejiggering old antibiotics, reformulating them, maybe making small modifications, but not doing anything that will bring entirely new structures or compounds into the market. So my hypothesis is that if we provide faster routes to discovery, because it's the discovery process that is so laborious and time-consuming and expensive, that would encourage some of the industry to get back into registering and marketing new antibiotics. And so that's been my goal for a couple of decades now. And in addition, to try to fight the myth that the soil is tapped out, as it's not. This myth has turned out to be the biggest barrier to funding. I never submit a proposal about this project without hearing, but the soil is tapped out. So overcoming this myth is going to be a really critical part to making this a viable field. So this led me, when I was professor at Yale, to develop the Tiny Earth program. There's really good evidence that research courses have a tremendous impact on undergraduates' persistence in STEM fields. Without any other changes in their curriculum, if an undergraduate takes a research course, they're far more likely to complete their studies in STEM than to switch to another major. And so this is an important way to keep students interested in science and to increase the size of the workforce, which is desperately needed. So I started, based on many other examples of this, I started a research course for antibiotic discovery. We call it Tiny Earth, or Student Sourcing Antibiotics from Soil. We maintain the pedagogy of this program across the world by directly training our instructors. In fact, the reason I couldn't be there in person today was that this is the week of training of Tiny Earth instructors in Madison, so I needed to be here for that. We now have a network of 700 or so instructors who teach. We estimate around 14,000 students per year. And we are now across the world, 31 countries, and most of the United States, and growing all the time. So this is our network for undergraduates who are discovering antibiotics by a really simple method that is tried and true. So the students choose their own site for soil collection, which is an important element in why we think we'll be discovering new antibiotics. They grow bacterial colonies on solid media. They isolate bacteria and then test them against a series of other bacteria or fungi in some cases. And then if they want to, they can pursue the chemistry in their own institution. But because many of the students go on to other courses, or they're at community colleges that don't have research programs. Many of them and their instructors choose to send their isolates to the Tiny Earth Chemistry Hub, which is in my institute, the Wisconsin Institute for Discovery, where we try to do the chemistry on the isolates that they sent us. So whenever I talk to people who have been in big pharma in antibiotic discovery, they laugh at me and they say, well, why do you think you're going to discover things where I couldn't? Because the soil was tapped out in my hands. And our strategy is a pipeline that emphasizes new sources by having the students collect their own samples wherever they want, which is not the soil sampling grid that the companies used. We screen against new targets. We use the taxonomy of the producer as a guide. We use genomic analysis and new screening methods. And I want to just briefly go through those elements of the pipeline. There are others, as you see here. So the first element that's new is when the industry pulled out, we didn't even know about, for example, Acinetobacter. Acinetobacter was nicknamed Iraqibacter because it started affecting troops in the Iraq War. And it originally came from the desert, but now it's pretty much ubiquitous. So the industry would never have found an antibiotic that was specific for Acinetobacter because that would not have been in their screening. Another element is we desperately need antibiotics that affect gram-negative bacteria. Those are highlighted in red here. And most of the antibiotics that they discovered worked against gram-positive bacteria. So again, we're interested in a slightly different range of compounds and activities. We also, in my lab, screen against oomycetes, which are typically plant pathogens. These are protists. Lower eukaryotes that produce these gorgeous structures and swimming spores. And in fact, the first antibiotic we discovered it remains of interest today. It's called zwittermycin A, a very unusual aminopolyol. You can see its inhibition of Phytophthora there. And finally, we collaborate with a lab that does anti-helminth screening, and this is for both human and animal parasites. So this is a variety of targets that were not typical in industry. Certainly, they didn't screen against oomycetes, and most did not screen against worms either. So we start with the antibiotic activity of the strain that the students have sent us. We also look, besides the target organisms being different, we also look for narrow spectrum antibiotics. The industry shooed those because they figured they'd be of even smaller market size than the typical antibiotic, and that they weren't worth pursuing. Now we know that narrow spectrum antibiotics are a great way to reduce the frequency of resistance. So we think those are worth keeping in our screens. We then sequence the 16S rRNA gene of the organisms, and we focus specifically on taxa that do not have any known antibiotics in the literature. So hopefully we're again optimizing the chance for discovery. We then do a genomic analysis in collaboration with the Joint Genome Institute, Department of Energy. They are sequencing about 500 strains completely, doing beautiful closed genomes, very high quality sequencing. That gives us the ability to begin to look for novel biosynthetic clusters in the genomes of these organisms. And there are a couple of really good elements of software. In fact, one of my postdocs is one of the developers of Anti-Smash that have the ability to ferret out these biosynthetic gene clusters. And starting with those is a great way to identify strains that look like they might have novel compounds. And then finally, we turn to chemical analysis. So this is just one example of a biosynthetic gene cluster that we found in one organism. You can see that this is the query sequence here. This is one that wouldn't interest us all that much because you can see that these are all below it are all the known sequences that are similar to it. Some of them, most of them with 100% identity to the query sequence. So that's one that we would quickly discard. But we also find much more often sequences that diverge quite deeply from the known antibiotic biosynthetic pathways. And we can predict, in some cases, the structure of even unknown antibiotics based on the genes that assemble the compound. This is an example of one of my favorite strains. It was a Pseudomonas that was isolated by a high school student at Glenbard East High School. Most of our students are actually in college, but there are a few high schools. And this is an example of the richness of the genomic information in these organisms. So these are all the regions of the genome of this strain TEC 758. And these are the types of compounds that would likely be produced by the genes in these regions. And you can see that some of these would not be interesting. For example, the anacasin pathway, 100% identity to a known non-ribosomal peptide synthetase system. So that wouldn't be of interest. But then there are others like the fengysin-like molecule, that's the closest relative, but it only has 13% identity to its best match. So that would be a beta-lactone, for example, that we might pursue. So just an example, a summary of a few organisms. When we did our first 22 genomes, we found 824 biosynthetic pathways in their genomes. That was just 22, and we're updating this right now. We have many, many hundreds of more. And you can see that some are quite common. We see a lot of what appear to be saccharide and fatty acid biosynthetic pathways, a lot of non-ribosomal peptide synthesis. But then we see some like the thiopeptides that always light us up. And when we find a thiopeptide pathway, we pursue it. Terpenes as well. They're much more rare, and there are fewer antibiotics based on those kinds of structures. So that's the next element of our sieve, is the genome. But all of these pathways are not necessarily expressed. For example, the organism that was found to produce streptomycin many, many years ago by Selman Waxman only produces streptomycin, but when its genome was sequenced, it turned out it had 26 other pathways that were all silent. So one of our biggest challenges today is to figure out ways to induce those silent pathways. And this, we are just scratching the surface on this. I'm just going to mention this one example of a very exciting discovery using sublethal levels of antibiotics to induce silent pathways. We started here, this was actually an undergraduate's project that came out in a way that has really shaped the rest of our work. She took 4 strains. One did produce a zone of inhibition against Acinetobacter. The other 3 did not, when plated on PDA, which is potato dextrose agar. When she did the same thing, but she added a sublethal level of streptomycin, you can see that 2 more of the strains produced zones of inhibition against Acinetobacter. So these are the kinds of strains that we're particularly excited about pursuing, because we know that the industry didn't use antibiotic induction to find antibiotics. So the combination of new targets, the taxonomy of the producers, the genome sequences that allow us to recognize new biosynthetic clusters, and then screening methods that include induction of silent pathways are maximizing the probability of discovery in our pipeline. And these are just a few examples that I won't have time to go into that have been discovered either in the Tiny Earth Network or in my lab. With that, I'd like to stop and leave some time for questions, and thank all the people who do this work, all of my staff, students and collaborators, and particularly the undergraduates who have worked in the lab, who bring an incredible level of creativity, which is one of our silent weapons to discover new antibiotics. Is we're not using jaded scientists who have all the biases of the antibiotic industry in place. These are young and fresh perspectives that we're applying to the job. And finally, the Joint Genome Institute, we couldn't do this project without their fantastic sequencing capacity, and we thank them for collaborating with us. Thank you for your attention.
Thanks so much, Joe. That was really fascinating. Do we have some questions for Joe? Kelly.
Hi, Joe. Thanks. I really enjoyed the talk. Good to see you. So I like the discovery aspect, and I agree that's the major hurdle. But in like, and I know there might not be answers to this, but I'm trying to think about how I'm going to write this because it's one of the sections I get to write. So you can help me out and give me some cuts here. Other hurdles that we should account for, right? So as you know, this is one really great observation and kind of a portfolio for enhancing the portfolio. But what are other ways if we want to kind of go back to this kind of era of bioprospecting? You know, what are some other suggestions or areas that we can do in your mind as somebody works in this space that I could include in the report in terms of bioprospecting and kind of overcoming this soil is tapped out phenomenon is one. It sounds like I resonated with me, but other ideas that, you know, you would like to see stressed in this report?
Sure. So one area that has been a problem for many years is the registration requirements for antibiotics. And generally they have to be held to the standard of the standard practice, which would be an antibiotic that works against a certain pathogen. But that's a pretty high standard, given that if the pathogen is resistant to that antibiotic, you certainly want the new one. And so I think we've been using kind of the wrong standard. We should be using resistant organisms as a secondary standard and say, okay, it may not be as good as ampicillin, but if your bug is ampicillin resistant, then you might want to turn to this one. So that's one thing. There's been some recent legislation, and I can study that and send you some summaries that has tried to encourage antibiotic registration, but it's still a really expensive and difficult process. And the difference, I'm told, between registering antibiotics and registering cancer drugs, for example, is simply getting through the regulatory system is harder for antibiotics. And people think if there's a new cure for an Alzheimer's drug or a cancer drug, they want to see it come to market. And so the process is much quicker. And I think there are ways that we could be making the antibiotic registration process faster as well. The next one is the Nagoya Protocol. And I was actually going to suggest to the academies that it might be time to write a report on the Nagoya Protocol. This was an agreement that maybe you've already talked about this, so maybe I don't need to go into it.
No, we'll talk about it, but then about that today. We had a long day, so
it was developed with really great intentions to prevent the bioprospecting and ultimate exploitation of particularly developing countries that have the majority of the bioresources in the world. But it became so strict and so restrictive that it's very difficult to get organisms out of other countries and into the US without going through a lengthy agreement process. And so the agreement that we have, we only take strains from US classrooms as a result. The agreement we have, it simply says, if there is something commercially viable, we will come back to you and we will negotiate a great agreement. And we have an excellent patenting and licensing arm for the University of Wisconsin. So they have a great agreement there. And we, of course, are good for our word. We want to benefit the schools that have contributed the strains for the benefit of the whole network. We can't do that because of the Nagoya Protocol. Now, the irony is the US hasn't signed on to the Nagoya Protocol, but we still think it's right to adhere to it, even though the US is not a partner, because its intent is so good. We have to figure out a way around that, because if we had to negotiate with every single country from whom we want to get strains, it would take forever in negotiating contracts. And so I think the Nagoya Protocol, as well-meaning as it was, has become a real inhibitor of innovation and collaboration. Those are just two. I'll send you some more by email.
Great. More questions?
I have one.
Yeah.
Hi, Joe. Yeah. I really enjoyed your talk. And one thing I've always really been so impressed with is, your interest in pedagogy as well as pedology, and the Tiny Earth program as a model for doing similar kinds of things to energize students around like maybe more broader soil issues. And the broad is part of the problem. But like around energizing around soil health, which unfortunately is so vague, in health with respect to what. And the beautiful thing about your target is it's much more clear, and I'm sure it's not easy, but it's clearer than the objectives for soil health. And so any thoughts about sort of mobilizing something like that? Because we've got the problem with soil health being very holistic and people being very anti-reductionist about how they want to think about soil health, battling against people who are, more reductionist in their approach. And obviously those findings will scale, in some way scale up or will help guide even more holistic approaches. But are there any thoughts about that?
Yeah, I have a real problem with the holistic approach because as scientists, it doesn't give us benchmarks to try to achieve. And it's very difficult for students if you can't give them a clear set of criteria. So I would love to get soil broadly into the curriculum. For one reason, a lot of high schools can't work with the organisms that we study. They can't culture from soil and all of that. It's just not safe. So if we developed a soil characterization program, that's research that most high schools could do. So I've been working with an engineer who develops biodegradable sensors, very, very inexpensive sensors. And he's trying to develop a whole series of them to measure aspects of soil health. And what we'd like to do is remap, we're going to start with Wisconsin, but remap the soils of Wisconsin. I have the most recent up-to-date soil map of Wisconsin hanging in my office, and it's dated 1968. So, and it's really crude. It's not very, very intensely sampled. So I think we could use this incredible workforce of students to gather a lot of data about our soils and finally have a better sense of the soils of the world. I mean, we have 22,000 species of soils, and we don't really have a clue where one begins and another one starts, and what the nature of the microbe heterogeneity is. So I think having a soil curriculum for both the college and high school level is fantastic. I totally agree. We need that, but we've got to get a little more precise than going out and assessing soil health. Use carbon content or other elements. We use aggregation as one of our measures. Give the students concrete things to measure, and they will do it for us.
Great, thanks.
Thanks. More questions? Oh.
I enjoyed the talk. Thank you. So I'm a soil ecologist and I'm not in the medical field, so this might be completely off base, but I heard somewhere with climate change that fungal diseases are going to become more prevalent. And I'm just wondering, like you said, you screen for, other than bacteria, some oomycetes, but do you increasingly screen for fungi as well?
We just haven't had the hands to do it in my lab. We just started using Aspergillus and Sclerotinia as a couple of plant pathogens that are interesting and that cross over to the human pathogen side as well. It's easier for the students to work with bacterial isolates. The oomycetes are we use Pythium, which is extremely easy to work with in the lab. So that's why we've incorporated that. And also I'm just interested in the uncontrolled aspects of Pythium. With all the research we've had on oomycetes for years, it's still probably one of the leading leading causes for crop loss, and we really don't control it very well. So those have been our top priorities just because they're easy. But yes, I totally agree. Fungi are important, both plant pathogenic fungi, because as climate warms areas that were not warm before and were intolerant to certain the fungi we're intolerant to, we're seeing those fungal diseases creep up mountains, for example, and expand their footprint because of the change in climate. So plant pathogens and animal and human pathogens, absolutely critical, high priority. And I should mention this, using the fungi as a source of antibiotics is another untapped area that we just haven't pursued, again, because we just don't have enough money or people.
More questions? Okay.
Oh, I've got one more. What about screening for biodegradation pathways? I know this is outside, what you do, but I don't know. We could have an effort on that as well, I suppose.
Yeah. One of the beauties of this course is that bacteria are so versatile that they can take on almost any challenge. And so a lot of our instructors have actually varied the curriculum. One of them does microplastics degradation, and they've actually found bacteria that look like they might be damaging some microplastics. And there are several other directions. And, if there's something particularly important in an area, contaminated river or, soil that's contaminated with a pesticide, that of course piques the students' interest because it's a local problem. So we've seen quite a few of them turn to those kinds of problems. Yeah, it's a great idea.
Rebecca has a question online.
Hi, Joe. I just wanted to thank you for a great talk and express my admiration for the sort of combination of massive research participation and massive education at the same time. I just think it's really brilliant thing that's underplayed in our research and education system both. So I just love it. And I wanted to ask you, often I think there seemed, perceived to be some big trade-off with regard to quality and quantity. When you go for big participation, you get, crap data or similar, but I was just curious what you would say about whether there is such a trade-off, or is it overwhelmingly gain over pain vis-a-vis quantity and quality of data when you engage people at that scale, which again, hats off to you.
Well, thanks. I think doing research with undergraduates is the easiest way to meld them together. And it also happens to be the most powerful, impactful way to change undergraduate outcomes. I think there always has to be a trade-off. I mean, you're using untrained until they take this course, unsophisticated scientists. You gain, of course, their innovation. When I taught the course the very first time, 11 years ago at Yale, I only had 6 students. Nobody at Yale was interested in this, except these 6 students. And they were just amazing students. And one of them, in the course of the course, decided she wanted to use this medium that you saw in one of the slides, PDA, potato dextrose agar. Well, it's the funniest thing to talk about PDA in front of a bunch of pharmaceutical scientists because they all say, what's that? Well, of course, I grew up in a plant pathology department, so I know potato dextrose agar is kind of, a useful medium. Well, it turns out that one of the most interesting things anyone's found in this whole network is the fact that you can double your rate of discovery of antibiotics by culturing on PDA. And we think it has more to do with the susceptibility of the pathogens that we're testing than actual production, but we're not sure. We're still working on why that is. But, that was in the first 5 minutes of the class. And so you benefit from that naivete. When the student first proposed it, I said, you really want to use that? I was as prejudiced as anyone in the pharmaceutical industry, but she went ahead and did it and made a great discovery. In terms of the quality, one of the things that's great is if there's a way of checking. And when you're taking measurements in the field, like the soil course that we were talking about before, which I think would be fantastic and I'd love to do it, you do need to worry a little bit about replication and quality of measurements. And that's why we're working on these very simple sensors that are very inexpensive. So maybe many students in a class could replicate each other's work. And that's a technique we use also, because the students are always wanting to conclude, there are more antibiotic producers in desert soil than in, rainforest soil, because they compare two soils and, with sample size of 1. So we try to get them to replicate across the classroom and have groups copy each other's work and then take averages, and then we can teach some statistics. So clearly we have to be careful when they're measurements. The nice thing about the antibiotic producers is that when they send us the isolates, we automatically have to retest them, not to check their work, but just to make sure that we have a way of assaying them. So in that sense, I think we're kind of protected from the quality issue. And there's just, the quantity is so enormous. We have so many isolates coming in from so many classrooms that, if some of them just don't work out, it's okay. But yeah, that's something that I think is really important to think about.
I'd love my time on those sensors, Joe, by the way. But anyway, that's great.
Okay, we'll talk as soon as we get those built. And the Christmas bird count is an example of citizen science, which has not had great checks and balances, except for the number of people doing it. And it's been incredibly meaningful data for decades. So that, I think, would say that you can use untrained people to collect certain kinds of data.
We have a question from Sam.
Hi, great talk, really enjoyed. My question was actually related to what you just mentioned, that soil being a source of compounds. My question is that you compare different types of soil, and I was wondering, are certain soils more rich with these compounds, or perhaps, for example, if the soil is pristine, does that mean that it'll harbor more of those, it'll have those compounds more? Is that, or is it like typical cropland soil or agricultural soil that, yeah. So when it comes to type of soil, yeah.
That's such a great question. We developed a worldwide database in the hope of answering that, where the students enter all their data. So we can't get their strains if they're in another country, but we can get their data. And we hoped that over the long run, we would accumulate so much data that we would be able to answer things like that, like what kinds of soils are the best yielding for antibiotics. We're not quite there yet. The database is maturing, and hopefully we'll have enough to answer that. But in fact, one of my students, a Native American student that came to my lab for the summer, wanted to compare continuous corn site that's been in continuous corn at a university plot for about more than 30 years with the Three Sisters soil from her reservation. So where they plant the bean, corn, and squash traditional combination. And she has found many more antibiotic producers in the Three Sisters soil than in the continuous corn. Now, again, sample size of 1, but very intriguing. So that's something that we're really interested in doing. We're just starting to compare prairie soils with agricultural, because the prairie soils of Wisconsin are just magnificent. They're incredible portraits of what soil can be. And they've been so degraded by crop management that it would make sense that there might be a greater diversity, a greater richness of organisms, including of antibiotic producers. But we actually don't know that for sure.
Thank you.
Any more questions? Okay, Joe, I hope you're going to stay with us.
I am.
We'd really appreciate it. And thank you very much for that presentation.
Thank you.
Our next presenter is Katie Karberg, who is the Medical Affairs Manager for Bayer Crop Science based in St. Louis, Missouri. She's a physician scientist that has worked in academia and industry in the sectors of medicine and agriculture, both in basic and applied research. The constant in all her work is the microbe world. How the microbes that live in, on, and around us impact human, animal, plant, and environmental health. She obtained her MD and PhD in microbiology from the University of Illinois at Urbana-Champaign with her research in microbial evolutionary genomics, especially how microbes co-evolve through horizontal gene transfer and genome reduction processes in complex microbial ecosystems such as the human gut. Prior to her academic training, she was a research associate in microbial genomics for an agriculture biotech company, Cirion Genomics, where she was seeking to harness the vast genetic diversity of environmental microbes, including soil microbes, for plant biotechnology traits. In her current role, she provides guidance on safety aspects of products for the entire product life cycle, from the discovery phase to commercialized products, with an emphasis on navigating the complexities of the human, animal, plant, and environmental microbe interfaces. Tapping into her diverse experiences, but also in the context of basic toxicology principles. Katie, the floor is yours.
Thank you so much for the invitation to speak here today about a topic that's so fascinating and so complex, but so important. I even had trouble coming up with a title for this talk because this is such a complex space. And so certainly can appreciate the difficulties in trying to put together a report on this. And I certainly hope that what I present today will provide you all with some useful information. The next thing I want to do is disclose my very obvious conflict of interest here. I'm employed by Bayer Crop Science, which is the agricultural division of Bayer, which a lot of people don't know that Bayer has an agricultural division, but it's a rather large division. And so what we do is to provide innovations for farmers. We sell our products to farmers from seeds, not just large row crops, but we also have a vegetable business with a lot of interesting breeding projects going on, as well as crop protection products and now digital solutions. So I'm going to try to touch on three very big topics today. I hope I can get it done in the time that we have for today. But certainly, hopefully, we can have some good discussion about this and love to follow up on any of these particular aspects. But for today, I'm going to talk about some of these interfaces, and there are many facets of interactions between all of these systems and the microbial world. And so in particular today, I'm going to talk a little bit about the plant microbe interface and how we can utilize that interface for new agricultural innovations. And then I'm going to talk about a couple topics related to the use of agricultural tools, which have impacts and interactions between the soil and human health. So with that, if I just show this simple view of an agricultural field, because certainly if there is an interconnection and a sort of pathway of connections between these different spaces, it would certainly be through the microbial communities that colonize every one of these compartments. And I just want to acknowledge that all agricultural activities have an impact on the environment. Just looking at this, and this is something that I think about as a nerdy microbiologist working on my garden. Every time I dig into the soil, I'm disrupting that microbial community, but I need to do that in order to plant whatever plants I want to plant. So when you look at this picture, obviously this is not what the earth has evolved in its native setting. On an evolutionary time scale, humans have been here in such a short amount of time that just by virtue of planting crops, which obviously we have to do for food production, it's one of the major advances in human civilization, you're inherently going to disrupt the communities, and particularly the microbial communities and various other communities in these compartments. And so I think that's an acknowledgement that just has to be done. But when it comes in particular to the microbial communities, we have to ask, of course, the question, what do these impacts, if we can measure them, what do they mean in sort of the larger scale picture of all of this? Because certainly every agricultural tool, no matter what we use, no matter what type of system, has benefits, but can also have drawbacks. And that we ultimately have to balance the need for the tools and the potential trade-offs posed by increasing the use of such tools. And so I always like to keep the big picture in mind because that's something that really motivates me. But ultimately, I think we have to find the balance between food production and not putting more soil and more land into production. And there's just one demonstration of that here. This is from our World in Data. And if you ever want to get lost in a bunch of data, that's a great website to do this. But they have all kinds of fascinating data sets to look at, including big sections on agriculture. And just want to highlight here, and I know it's a little small, so hopefully you can all see this, but this is the change in cereal production, yield, and land use and population from about 1961 to 2021. And this is just for cereals in this case, but this is actually the most important, the largest amount of crops that are produced. And you see cereal production increasing since 1961. And I would say prior to 1961, it's actually pretty level in terms of, all of these food production metrics. But you can see cereal production has increased pretty dramatically along with yield and along with the population. But you see that the land use has been pretty, we haven't put, a ton of land into production to account for these yields. And of course, yields aren't everything. I think, everyone in here is well aware of that, but it still is a major part of what we have to do to anticipate the needs to produce crops with a growing population. And I think one of the most important aspects of this, which I already alluded to a little bit, is by increasing production since 1961. And I will say that's through multiple kinds of innovations. It's a big part of that is breeding and crop breeding for better traits. But it's also due to synthetic pesticides and synthetic fertilizers, as well as mechanization advances that have allowed for these increases. But ultimately, with these increases in the uses of these technologies, we have to think about the land that has been spared had we not had these types of technologies on board. And that is demonstrated here in the brown as opposed to the green, which is the land in production, and brown is the land that has been spared. And you can see it's substantial. So what 1.75 billion hectares of land has been spared. And as I mentioned, we have to anticipate the global population change and what that means in terms of land, arable land available per person. So whereas we had about 1 acre of land per person available in 1961, by 2050, we anticipate this land to account for the world population increase will have less than 1/3 of arable, 1/3 of an acre of arable land per person available. So we've made these tremendous strides, but we have to do better than that. And when you don't have certain agricultural tools on board, the outcomes are dramatic. Without synthetic fertilizers and pesticide and other innovations, it's not just synthetic pesticides and fertilizers, crop losses can be huge, 40% to 80%, depending on the type of crop. And you can see here just fertilized versus unfertilized. And this was with early applications of phosphorus. But we know that there are negative outcomes from using these types of innovations. So this is just a schematic of the dead zone in the Gulf of Mexico here at the Mississippi River Delta. And this is one of the main contributors of this is actually from a nitrogen runoff from agricultural use. 59% of nitrogen inputs actually are lost to the environment, which is huge. And of course, this doesn't make growers happy either. They don't like paying for inputs that just get lost to the environment. It's not good for them. It's not good for the environment. It's not good for any scenario that you look at it. And so the bottom line is, can we do better? And we have done better in the last 60 years. And I think there's so much hope for doing better and using particularly the knowledge of the microbial world as a new step in innovation. And so when we think about the plant microbial interface, I think this is a particular area that we hoped that there will be a significant amount of innovation to try to offset some of, for example, can we offset some of the use of synthetic inputs, particularly in the biofertilizer space, so we don't have to use as much synthetic fertilizer. And so there are several different microbial compartments to think about. The phylosphere is the above ground microbial habitat. The endosphere is the microbial habitat in interaction with the internal plant tissues. And the most important, I would say, I guess I should be careful what I say, but I think the most active area for microbial plant interactions is in the rhizosphere, which is in the soil near the roots. And so in particular, this is there are areas of innovation in all of these spaces, but the rhizosphere in particular, I think, will become more and more important. And of course, there's many different types of microbial interactions. There's, of course, plant pathogens, viruses, fungi, and bacteria that plants have to deal with. And then there's commensal organisms, and that's a little bit vague. It's basically, there's microbes are there, but we're not sure what they're doing. They don't seem to be harming the plant, but they don't necessarily seem to be helping the plant either. But it may be we just don't understand those interactions just yet. And of course, there's beneficial microbe-plant interactions, which I'll talk about a little bit more. As you can imagine, it's a really complex space and a lot of interactions and symbiotic reactions going on between both the microbe and the plant. Of course, my rhizosphere microbes can help with nutrient solubilization. They can actually confer some pesticidal traits to help combat different soil diseases. There's some interesting data out there that can demonstrate that there's some potential for them to help by virtue of biofertilization and their other sort of stimulation of plant defenses, and also providing some pesticidal capacities that they can hopefully increase yield. And so, there's a lot of different influences here, all which factor into, how they need to be tested and developed. But I want to call out the fact that soil microbes have actually been intentionally used for well over 100 years in agriculture for both biofertilization and biopesticides. I think the first patent that I'm aware of for use of a soil microbe is actually from 1896, and it's for a sort of a nitrogen fixing bacteria. And so, this has been around for a long time, but the capacity to scale this to useful, scalable uses, I think now is just coming to be realized because of our capabilities to understand these interactions better. And of course, for biopesticides, Bacillus thuringiensis has also been around for decades. Used in organic agriculture, it's also the basis of Bt insecticidal crops. And there's expanding development for all types of different applications, for foliar, for seed-applied microbes, and for in-furrow uses. And that's all across industry, both small and large companies, and of course, academia as well. Now, a few factors about microbial products for agriculture, and I thought this was kind of an interesting, when I heard about probiotics and their impacts on the human gut microbiome, one of the things that was discussed previously is that colonization is not the only important factor. So it may not just be the presence of the microbes and colonizing that environment, but probably what's just as important, if not more important, is the actual products that microbes are producing, such as lipopeptides with antifungal activity, bactericides, plant growth promotion molecules that enhance nutrient uptake, as well as stimulating the plant immune system. Those might be the most important components, but kind of connecting to what Joe just said, environmental conditions play a huge role in production. You might have that capacity in a microbe to generate some of these, what we see as beneficial products, but you have to have the right environmental conditions to get these products to work. So it's not just as simple as adding a microbe and hoping that it'll work. You have to have the right environmental conditions. And so there's a lot of work ongoing in field trials to understand, what are some of these environmental conditions that can stimulate these beneficial effects. And of course, too, one of the bigger questions is, can microbial interventions impact plant nutritional quality? And I'm actually going to talk about some recently published work from some Bayer colleagues. This is not my work, it's their work. But in this particular study, this was a study where a microbial or a microbe was added in a spore form to the in-furrow to potatoes. And this was a rather large study, over 147 sites.
using 50 farmers and different potato types. And this was a several year study as well, about 4 years. And what they did is they added this spore formulation of microbe, it's Bacillus amyloliquefaciens, to the potato, to the soil where the potatoes were planted. And then eventually a symbiotic reaction or a symbiotic relationship was established in this environment. And a really big question is that even if we can see beneficial traits or beneficial outcomes from these particular studies, what does this mean for human nutritional impacts, if anything? So there were several different aspects of the study that were quite interesting, but I'm only going to call attention to this, the finding here on the relative nutrient content of these potato tubers after microbial treatment compared to standard farmer practices. And the green on the left there, these are the sort of the macronutrients and the secondary nutrients, and on the red there, those are the micronutrients. And you can see that there's some relative increase, in particular for iron there. Now, I want to be clear, what does this mean from a nutritional standpoint, a human nutritional standpoint? This is just a relative nutrient content. They don't have the calculations on absolute nutrient content, but I would expect that this is not sufficient to actually have convey a nutritional benefit for humans or animals either. And so, you know, that's okay though, because maybe what the benefit of these types of interventions, if it can't get to the point of actually enhancing nutrition for human uses, it's still of use to the plant and it's of use to the entire system, that this is a better way to possibly mobilize nutrients and again, decrease the amount of, you know, various other inputs, including synthetic inputs. So I just want to make sure that to level expectations there. And the mechanism by which this sort of nutrient uptake occurs is that the bacterium actually stimulates growth of these smaller types of roots, which really allows for more surface area for enhanced nutrient uptake. But I want to also explain a little bit, provide a little context on how complicated it is to actually understand nutritional outcomes. So we're going to switch over to soybeans in this case. And if you look at, this is a single genotype of soybeans, and you can see in this 50-acre, this is a 50-acre field here, and you can see the difference in protein percentages here that are entirely dependent on the different environmental compartments of this plot. So it's probably in large part due to different soil types and any other types of inputs that can influence the growth. So it's really complex, and it's not as simple as, you know, you can have, you know, it's not just plant genetics. In other words, there's obviously a lot of complex interactions that can lead to different nutritional outcomes. This is something I think, you know, we have to keep in mind. There's a lot of promise in this space in terms of using microbes in agriculture, but there are still a lot of challenges, many of which I think are tractable, right? I think innovation and research can actually get past a lot of these outcomes. But I think we also have to have some realistic expectations of efficacy. Will these microbial interventions will be able to completely replace synthetic inputs? I don't think so. I kind of think about this in the sort of, you know, how we treat certain human diseases. So like hypertension or diabetes, one of the first things that a good practitioner should do should suggest lifestyle interventions, you know, healthy diets, better exercise. And I think in some way we can think about microbial interventions in agriculture in a similar way, that this may be a way to sort of manage earlier, you know, sort of an additional tool to help prevent more severe conditions. But there are a time and a place where you have to apply certain medicines, if you will. And I think, you know, for intense insect pressure, that's a case where I'm not sure that, you know, we will be able to get rid of synthetic inputs, especially. The other key thing, though, I want to talk about, and this also relates to what some of what Joe was just talking about. I think one of the biggest hurdles for this space is actually the regulatory uncertainty. It's not really clear yet how different regions will regulate microbial products, biopesticides versus biofertilizers. And furthermore, there's a lack of regulatory capacity to handle all these innovations that are coming, as well as fit for purpose regulation. And that's a really big deal for any company because, you know, what they like to understand and see is regulatory uncertainty. There are far more innovations that sit on the shelf that ever get commercialized. And that's because there's not sufficient funding, even for big companies like Bayer, to get all of these products through the regulatory system. So I think that's a really important space of investment and development is to does this problem get circumvented? And that's not something that we on the development side can actually do. I also want to call attention to, there's a lot of regulatory uncertainty also for this concept of bioengineered microbes. So for example, we know that legumes have associations with nitrogen-fixing bacteria, but other crops don't quite have those associations. So what if we can take like a microbe that colonizes corn roots, for example, and we can engineer in nitrogen-fixing capacity? So it's certainly technically feasible, but it completely changes the regulatory pathway in certain regions, which is again part of that regulatory uncertainty. I actually struggle with this as somebody who's thought about the microbial species concept a lot. We know that horizontal gene transfer is rampant in the microbial world over vast phylogenetic distances. And so, you know, when we're thinking about bioengineered microbes, I think that's you know, something that I struggle with. How do we define whether it's bioengineered or not? If we don't know in a given microbial species yet, we haven't yet understood the microbial or the genetic diversity in that species. It's something to think about. But again, we still don't have a biologically relevant species concept for microbes. So I'm going to shift gears now a little bit. Hopefully I'm not too far out of time here. But obviously another really big connection, I'm going to shift away from plants for a moment and talk about the soil-human microbial interface. And as Joe mentioned earlier, antimicrobial resistance is clearly one of the most important connections between soil health and human health. And I would say, as somebody who has worked in the hospital, and I think any medical provider who has ever worked in a hospital setting has lost a patient to this issue. And I wholeheartedly agree that it's only going to get worse. So we have to think very clearly and strategically on how we combat this issue. And I want to talk about this just very briefly because I remember in a past session, there was this, there was a presentation about detection of antimicrobial resistance genes in an environment, and how do we sort of tease out what is, sort of an area of concern or risk that you want to think about, and perhaps institute some mitigations versus antimicrobial resistance genes that are just resident in the natural setting. Because of course, antimicrobial resistance genes are ancient. And with the use of antimicrobials to treat infections in human and animal medicine, these genes have been mobilized to pathogens. And I think an important key distinction between environmental antibiotic resistance genes versus that of which is in pathogens is that the pathogen human, that are of clinical relevance are located almost across the board within mobile elements. And mobilization of these genes represent a significant risk factor for high-level resistance expression and for further dissemination. So when you're trying to tease out what is antibiotic resistance of genes of concern in the environment, that is just resident, I think genomic context is a huge part of teasing out sort of relative hazard. So just as a really high level schematic, this is an environmental bacterium that has an antibiotic resistance gene that is chromosomally encoded. And then this is the next sort of scenario where you have this same gene But the little yellow boxes here are meant to represent IS elements or insertion sequence elements, which is associated with the beginning of mobilization of these genes or some aspect by which that they can then, it's like a stepwise sort of process by which they can become mobilized. And then you have the scenario where these genes can be mobilized to elements such as plasmids. And these are genes that are much more likely to be disseminated than amongst other bacteria. So we can assign sort of a relative hazard scenario here in a given population. But of course, there's the other element that we have to think of here. It's not just hazard, but it's risk is hazard times exposure. And so there's a lot, I think, potentially, if we think about mitigations, we can think about the exposure aspect of this as well. Now, it's really hard to assign risk values to any of this or even to the hazard sort of conceptual framework of this because it's just an unknown quantity. And the other thing is that once you add antibiotics into the mix, that certainly is a strong selection pressure that can take something that's relatively low hazard and then change that to a much higher hazard scenario. And again, there's obviously some interaction between environmental bacteria, human and animal commensal strains, and also pathogens. But certainly here, the higher risk scenario in an environmental compartment would be a human or animal pathogen that has one of these mobilized genes. And obviously, it's a lot easier to take a single isolate and then sequence it and then look for its, do a transferability assessment. But I think as our metagenomics capacity expands and gets better and we have longer sequence reads, we'll be better able to use these large data sets to kind of ascertain a relative sort of risk scenario for the detection of antibiotic resistance genes in the environment. I also want to go back to the topic of antifungal resistance. Certainly, this is a growing problem. I think in the clinic, there's certainly more fungal infections not only showing up in the clinic, but also ones that are resistant to our antifungals. And we don't have a lot of antifungals that we can use because they're typically pretty toxic. But we also use them in agriculture. And there's, certainly a lot of discussion and dialogue on what do we do about this? We need antifungals in human medicine, and we definitely need them in agriculture. Don't have time to talk about this today, but fortunately, there's a great resource that you can use. There was a workshop actually held last year on the role of plant agricultural practices on the development of antimicrobial resistant fungi affecting human health. So this is, I think, if you're not aware of this resource, this is a helpful resource to kind of inform people of the problem, but also potential mitigations and steps forwards in this space. And then I want to talk about very quickly here, I think this is kind of an elephant in the room, and we see this a lot on the agriculture side. There's a lot of interest on, what are the impact of different agricultural tools, especially pesticides, on complex microbial ecosystems? And does this translate to health outcomes? Again, this is a very complex topic, but there was an FAO publication that came out actually in May of this year talking about this topic. And I think, really when it comes down to assessing the complexities of a microbial community and any given input, I think what everybody is trying to understand, are microbes sort of the, kind of canary in the coal mine, or is this, not being able to see the forest for the trees sort of situation? And I don't think anyone knows at this point. But what I can say is that, it's clear what the gaps are in trying to do a risk assessment in this space. And the FAO publication highlights this very nicely, is that, still after all this time of research and development in this space, there's still a lack of a consensus definition of a healthy microbiome. That certainly applies to the soil as well. And so there's a real need to define microbiome relevant alterations and microbiome derived adverse effects. And then, of course, what's really essential for any sort of risk assessment is identifying suitable biomarkers. And then, of course, you have to establish conditions under which biomarker values are considered either normal or abnormal. And then really essential is to establish causality from a given perturbation with an adverse health outcome. The other key things, which is essential for sort of any regulatory testing, is standards. And currently, that is completely lacking in the microbiome space, no matter what type of microbiome space you're talking about, whether it's the human, plant, or soil microbiome communities. There's a lack of standardization for both in vivo and in vitro studies. So a huge need there. And I'll have that linked in the references that I include. And again, the National Academies has also taken a good look at this as well. And that study was published in 2018. So that's another resource, I think, that also highlights many of the same issues and potential step forwards. So I refer to this all the time. So a really important resource. So when it comes to pesticides and the impacts on human gut microbiota, for example, we don't actually directly study microbial impacts to microbial communities for all those reasons that the FAO reports highlights. In particular, there's no standards, and you have to have standards to have meaningful sort of data outcomes. But it's not as if this just that it's completely not addressed. So when we look at the standard hazard assessment for pesticide, and this is sort of the bare minimum of what it takes to register a pesticide, this is the amount of studies here. And this is something that takes years and tens of millions of dollars. So yet again, part of the complexity of getting a product registered. And I'll just zoom in on the animal toxicological testing, which is very extensive from acute, subchronic, and chronic effects. And some of these chronic studies here that I'll highlight there, these chronic studies are, through the reasonable lifetime of the animal. So in the case of the rat studies, you study rats for 2 years, because after 2 years, then they begin to develop adverse health outcomes of aging, such that the controls no longer give you good information. But these are studies that are done in animals that have intact gut microbiota. And they are exposed on a daily basis. So these are chronic oral exposures. And so if the microbiome is the conduit through which adverse health outcomes are mediated, in theory, you should capture that through these chronic studies. And the way that pesticide safety works, or the way that regulators set safety levels, is that you take the most sensitive endpoint from all of those animal toxicological tests. You find the most sensitive endpoint, which is then called the no observable adverse effect level. And then you add additional safety factors of 100 to 1,000, depending on the particular hazard profile of that pesticide. And that becomes the total allowable daily intake that regulators say is safe, at which you would expect no unreasonable adverse health outcomes. But of course, the FDA monitors dietary pesticide residues. And what we know about actual pesticide residues is that they are far, far less than the allowable daily intakes. So there was a study done in 2019 that was published, and that resource will be in the references as well. And in a study that looked at 300 pesticides in the FDA database, they could only get detections for 77 of those pesticides. And keep in mind that these are very sensitive analytical chemical assays. So they can only get detects for 77 of those pesticides, and all of them were less than 3% of the ADI, and most of them were less than, there was a 1 million-fold safety factor from the allowable daily intake. So I think that's a really important consideration as we think about possible pesticide impacts to the human gut microbiome. Of course, Paracelsus said all substances are poisons. There is none that is not a poison. The right dose differentiates a poison and a remedy. And then I just want to put this all into context. There's a great study in PNAS from 1990 by Bruce Ames, who actually came up with some of the toxicological testing studies that we still use today. And this study, this is the title, it says dietary pesticides, 99.99% all natural. So yeah, I love this one. So 99.9% of the pesticides in the American diet are chemicals that plants produce to defend themselves. Of course, plants can't get up and run away from different insults, so they do produce a lot of pesticides. Only 52 natural pesticides have been tested in high-dose animal cancer tests. Of those, 27 are rodent carcinogens present in many common foods, including many of the healthy foods that we should all be eating. And then Americans eat 1.5 grams of natural pesticides per person per day, which is 10,000 times more than they eat of synthetic pesticide residues. So again, there's certainly even a hazard in the fruits and vegetables that we eat. And so just because that hazard is there, should we not be eating fruits and vegetables? Absolutely not. All of the epidemiological data strongly supports that a diet rich in fruits and vegetables leads to positive health outcomes. And so I think it's really important as we think about this space that we put this all into context. Does that mean that we should just rest on our laurels and not worry about it? No, of course not. The other thing about pesticides is that the registration of pesticides is only good for about 15 years. And then there has to be a whole renewal process that looks at any data in the literature, including, public literature that would indicate that there's something to be aware of. Now, when it comes to soil microbial communities, there's less that's done in regulatory testing, I would say, than what we do on the animal safety side. But again, it kind of, comes down to how in the world do you test this? How do you have meaningful data that can actually inform, what are the best next steps? And I have no doubt that agricultural activities will have some impact on soil microbial communities. Again, plants root exudates will recruit different types of microbial populations based on the different chemistries that are present. But obviously, there's a great need to understand how this impacts overall soil health and functioning in the bigger picture and in the context of how to scale solutions that optimize crop production in a more sustainable way. But there's a little bit of hope, and I promise this is my last slide. I do think there's a lot of hope in this space. There has to be hope because we have big challenges, but we've got a lot of bright minds in this world. I think that can bring forth important solutions. So much like medicine, there's a lot of innovation happening in precision agriculture and actually giving farmers the tools so they can make prescriptions for their individual land and for their individual circumstances. So this is just a screen grab of a farmer's field with different soil types there indicated by different colors. And there's a lot of studies ongoing. Bayer's doing a lot of studies, but many other companies and many other academic studies are all doing studies on, how can we better use many different data inputs to better inform the different inputs. And this is just one example of these types of prescriptions. So you can see based on the different soil types, you might need to add a certain amount of nitrogen fertilizer. And I think there's tremendous hope in terms of, as we learn more in the microbial space, how do we also integrate these types of prescriptions for these environments? And so any input, I think, as we learn more, can be integrated into these types of solutions. And I think, as far as I can wrap my mind around right now, I think this will be the best path forward because these are such unique ecosystems. And there's so many covariates that can impact how a farmer has to grow their crops. And so I really think we'll see a continued expansion in this space. Just summarizing everything that I mentioned here, and I'll just skip over this for the sake of time. I don't know if we still have time for questions, but happy to take them. Thank you so much.
Thank you very much. This was a big scope of information that we had questions about earlier this morning, so it's really great to hear this. I think we're going to take our break and then hold questions to the end.
Yeah,
You're going to
sounds great.
stay,
All
right?
Yeah, absolutely. Yeah. I'll
Okay, that'd
there.
be great.
Thank you.
Yeah, so we'll take a 15-minute break and then come right back. Welcome back, everyone. We will now hear from Bhimu Patil. He's the Regents Professor, inaugural Leonard Pike University Professor and Director of the Vegetable and Fruit Improvement Center at Texas A&M University. And he's the Director of the USDA National Center of Excellence for Melons. Dr. Patil's research focuses on the profound impact of fruits and vegetables on human health, something that came up in our previous presentation. And he has particularly explored the effects of harvesting, post-harvesting practices on the food quality. He's pioneered the concept of foods for health, integrating various aspects of fruit and vegetable production, including breeding, agronomy, harvesting, distribution, sales, health benefits, quality assurance, and food safety, with an emphasis on consumption. Thanks very much for being here with us today.
Thank you, Dr. Wall, and all the committee members for inviting me to give this presentation. And I hope I'll be able to justify the presence of here to talk on the soil, because I'm not an expert on soil. But we still do a lot of research related to the field operations and how the crop production management, and the breeding, how they impact on phytochemicals or human health properties. So I'm going to basically touch on those things. So my outline is, again, I may not cover everything what I said in the outline, but I'll try to touch some of those and I'll skip some of those. So I'm going to talk about current health situation and where we are now and where we're heading. And also what are the facts of soil and what are the connections between the soil physico-chemical properties and how that impacts, again, phytochemicals. And also the soil composting and soil microbiome. And I think my favorite topic is how these soil properties will impact on human phytochemical or health promoting phytochemicals. I'll use few case studies where we do a lot of studies on different crops. Hopefully that will help to understand how the soil plays on human health properties. So this is the way we are, we have been. I think we are bombarded with this fast food restaurant Coke. And of course, I probably drank Coke after 10 years today because I was looking for water, but I'm also one of the culprits on that. So this is an experimental kind of burger came into the picture. We are bombarded with all this kind of food. This is the American diet. I'm not going to say everybody eats this. I think that's why I put that historical perspectives and how this our food is changing. At least now we're realizing the healthy food a little bit in the last 10, 20 years. So you can see that how we are evolved for the traditional diet to now. So we were depending on fruits and vegetables earlier. We were hunters and gatherers, and then we started eating a lot of carbs. And then we got into this stage. So while we're dealing with the obesity, on the global level, we're also addressing the malnutrition. So these are two major challenges in the global level to really fix the problem. So again, global obesity is going in not just the United States. A few years ago, I used to give a talk in other countries. I thought it's only the United States, but it's all over the world, the obesity is increasing. Having said that, of course, the United States is really on the top in the obesity ranking. So with that, okay, is your food is safe? Again, as all of you are discussing in the last several weeks or months about how is this soil and some of these effects of the soil, like even contaminated water impacts my food. Is it safe is a question, right? So these are some of the facts. Of course, as we all know, and I think I watched some of your videos, soil is really dynamic. Of course, while the soil is dynamic, it has an image issue. People consider it as a dirt and germ. And I think the previous speaker, Joe, really were doing a great job on educating the kids about this soil and how the health situation, because when 15 years ago, when we tried to recruit students to our labs, they used to think all agriculture is mud and dirty. So at least now the perception has changed. We're not only dealing with the dirt and mud, but we're also dealing with human health properties. So now the students are coming to the labs and doing a lot of research in nutrition and animal science and other stuff. So again, I'm not going to read these details, but I think we are impacting the degradation by 1/3 of the farmland is already affected, expected to degrade about 1/3 of the by century. To get 3 millimeter of soil, it takes a century, right? So this is the challenge. It's a complex interaction between the soil and human health. Of course, this group has been spending a lot of time on soil microbiome and human gut microbiome. There's a strong linkage. But a lot of the other characteristics of soil, for example, organic matter, chemical pollution, how they really impact on different human health issues, even skin disorders, cancer risk, as well as respiratory issue is a big thing. So there is a lot of interlinking between different factors. So if this group address this issue, I think we're really giving a lot of great move. I don't know whether we can fix all of these in this coming 10 years or 20 years, but there is a lot of things to study. I think there's a lot of interlinkage there. So I'm going to touch on one or two factors here. I'm not, because of time, I'm not going to cover on the climate change and human interference, but I'm going to touch on how the soil physicochemical characteristics and the microbiome affect the phytochemicals or human health properties. So to touch on the soil salinity and stress, for example, there are several examples here. Spinach is one of the examples. There are some genotypes which are resistant, and they can increase, they're resistant to salinity, but because of that resistance, you can have higher levels of the phytochemicals. And similarly in lettuce, you can, salt resistance will help in increasing the post-harvest longevity because of the reduction in the cell size and larger number of cells. So these kind of advantages are there, and some of the, even the salty soil, which sometimes we kind of think that salty soil is not great, but for certain cases, salty soil is better than fresh water for certain crops. So coming back to how these essential elements play a role on, again, phytochemicals, soil is made up of about 45% of the minerals. So these mineral function under the 4 categories. One is building blocks of their plant metabolism, and they're also good regulators of the enzymes, and also antioxidant. And the stress sometimes is good or bad to the plant, but it will increase some of these phytochemicals or plant defense compounds. So best example is iron. I think the previous speaker touched on that. So on the plant, definitely it causes chlorosis, but on the human health, iron deficiency is a bigger issue. So for example, here, currently with the new recent data, about 30% of the US population, both men and women and children, are iron deficient. So coming back to the microbe, how they, soil microbiome, how they affect the health promoting compounds. So this is one of the examples. Soil microbiome, of course, this group has been already discussing a lot, so I'm not going to spend a lot of time on that. But just to let you know that this has a good impact on structural integrity, which has a synergistic effect. Because of that, they regulate some of these hormones, and then they develop or biosynthesize some of those secondary metabolites, which in turn provides some of this good protein and enzymes and volatile compounds also. So this is in a small study as a part of our big collaboration, which I'll talk in a minute. But this is one of the students in University of Arizona, where Dr. Kerry Cooper's lab, she's just defending her thesis soon. So what she did is she collected soils and the melon samples on 3 locations, one in California and 2 locations in Arizona state. You can see the diversity of the soil particularly on the left side is the soil samples, on the right side is the melon samples. There's a complete difference in the soil microbiome attached to the plant as well as to the soil. So if you look at the significant difference in Shannon diversity based on the alpha diversity, you can see that the soil has a significant effect as well as the type of the location. It's a significant effect. Again, this is not completely done yet. That's why it is not published, the research. But this is an example how the soil microbiome impact on the crops. So this is another debate going on nutrient density. So for example, the current definition of nutrient density is basically based on the only food and how many calories. Because there's a debate and there's a lawsuit by some of the food companies. For example, Kind Food, they sued the government. Basically, okay, if you're talking about healthy, you cannot use nuts, for example, in the Kind Nuts product. That means it is more calories, but it is also higher in fat. So that is not considered as basically healthy food. Because of that, now they're changing this hybrid nutrient density definition. So the definition is there. Basically, you have to not only consider nutrient and the type of food in that. So that way you can really use that in a good nutrition policy and better public health. So with that, having said that, my talk will be on how different agriculture practices. There are three major agriculture practices. One is conventional, and another one is organic. We have been doing it for a long time, both. And then now in the last few years, regenerative. Basically, regenerative is basically kind of a modified organic with methods which can help to reverse the soil by using climate change. How do you adapt to the climate change? So if you look at this particular slide, there are a lot of literature showing that conventional agriculture is really reducing some of this. When I say nutrients, it's not just only nutrients in this case, it's also the different phytochemicals. And also it is really increasing the high levels of pesticide residue and antibiotic resistance, which is already alluded in the previous two speakers. So when you look at the next slide, it's basically organic agriculture. In this case, you can see that majority of the studies have shown higher levels of health promoting phytochemicals and some of these mineral elements, including the vitamin C and different elements also. And you can see that, again, carotenoids have increased. These are all basically based on organic. The top slide, or the top picture, it shows basically regenerative agriculture. You can see that different 5 crops, and again, higher levels of the vitamin C and other phytochemicals and some of the mineral elements. But in the bottom table, it shows if you compare organic and conventional, in most cases, only organic is showing higher levels of the phytochemicals in this slide and even the next slide. There's a lot of cumulative evidence showing that organic and regenerative agriculture is probably better for particularly human health properties. I'm not saying that we should completely forget conventional agriculture, but at least for the human health properties, that is really showing better results. So with that, I'm going to touch on some of these case studies, which we do as Dr. Wall mentioned, I deal with a lot of this farm to the clinic approach. When I say farm to the clinic, we call us table to the farm. Table to the clinic is basically in the future, the table dictates what the farmer has to do. When I say table, it is a consumer. So that's the way the future is. I think it's not it's not farm to the table, it's the table to the farm. So if the farmers do not understand what the consumer wants, I think the agriculture will fail, in my opinion, in future. So we have to understand what the consumer wants, because they are the one who dictates what type of food they want to eat. So these are some of these case studies. So we study a lot of the bioactive compounds or phytochemicals we call, of course, we call as phytonutrients. So when I started researching these citrus phytochemicals about 15, 20 years ago, when I used to give a talk, I used to ask questions to the audience, okay, what is it in citrus you like it? They say vitamin C. A lot of people don't know any about the other compounds. I'm talking about consumers. Because finally, at the end of the day, we have to convince the consumers, right? Whatever we do in agriculture. So when you look at these, there are about 7 to 8 categories of compounds. So there are about 500 phytochemicals, including the volatiles, are present in citrus alone. So that means if you take carrot or other examples, there is a lot of compounds. Do you know anything about how the soil impacts to these? We don't know yet. We're just scratching the surface. And about these 500, how many of the compounds we know are really beneficial, we still don't know. How many are there related to flavor? How many are related to some of the antimicrobial properties? So we're still trying to understand some of these roles. So what we do in our research is we isolate these compounds, we characterize, and we do in vitro studies, and we take in vivo, and then we work with the collaborators in clinical level. So these are some of these compounds we isolated from citrus alone. And of course, some of them are from turmeric in the last one. And some of these flavonoids on the left side are mostly citrus. Some of these limonoids and flavonoids. So these are the ones we isolated in our lab. We, of course, tested these in vitro in cancer cell and other in vitro and in vivo studies, which I'm not going to spend a lot of time in that. But this is just an example how the soil affects. We tried to grow grapefruit in Florida and Texas in this case. And if you look at the numbers here on the October to June, where we eat grapefruit, the previous months is basically not the right time to eat grapefruit. We grew in the same rootstock, except the soil were different. You can see the lycopene content is significantly different in Florida compared to Texas. This is an example of how the soil affects. So this is another example. We deal with onion. I think you must have heard Texas A&M is very well known for 1015 onion. We have contributed to Texas economy about $3 billion because of one onion, the 1015 onion. So because this onion was popular growers are trying to grow in different locations. This is a study where within about like 10 miles apart, growing different types of soil, you can see the quercetin content is very different, significantly different in about at least 5 genotypes and 1 with the variety. The last one is Texas 1015. That's the one which is popular in Texas. So this is another study in UC Davis, 10-year study, again, having this organic compared to conventional. You can see on the top is conventional, and the bottom figure is flavonoids content. And you can see that organic over the 10 years period is almost double in quercetin and camphorol and naringenin in both studies. This is what we did is when we think that the grapefruit has a lot of these phytochemicals, we didn't want to stop there. We thought, okay, what happens to these compounds? So we irradiated the grapefruit. The reason we irradiated the grapefruit is to ship the grapefruit from Texas to even Arizona. It has a quarantine measure because of Mexican fly and fruit flies. So we had to treat these fruits as a post-harvest treatment. So we thought, okay, when you treat, normally we show that, okay, there's a lot of bioactive compounds, but then what happens to the colon cancer in particularly treated fruits? So this is one of the example, different compounds only in case of flavonoids, hesperidine and naringenin and epigenin. The next slide shows you the grapefruit pulp and the irradiated grapefruit pulp. Interestingly, when you compare to the basal diet to all the other compounds, including the irradiated grapefruit, you can see the aberrant crypt foci is the colon cancer model indicator. Basically, you can see the significant reduction in those ACF or high multiplicity factor ACF in both all the diet, including the whole fruit, as well as the different compounds. So again, there's a lot of accumulative evidences are here on preclinical evidences on flavonoids and colon cancer prevention. That is, again, I'm not going to read those specific details. These are coming from the literature. There's enough literature to show that the flavonoids, which I showed in a few minutes ago, have a significant role in reduction in colon cancer. And these are clinical evidences showing that flavonoids have some significant benefits on colon cancer prevention. So what we did is we, again, as I said, we didn't really stop in one cancer model. We took these compounds we purified earlier. I showed that. So we have shown that these citrus phytochemicals, and including limonoids and flavonoids, you can see that it has significant reduction in colon cancer as well as breast cancer, neuroblastoma, and prostate and pancreatic. Majority of these studies are in vitro. Few studies are in animals. The question is always, is this really relevant in humans? Is a question. We don't have the capacity through USDA, maybe NIH can give money. So we didn't do a lot of the human clinical trials, but we have one human intervention study, which I'll show it in a minute. So this is, again, a good evidence of literature playing flavonoids in cancer and how these specifically flavonoids play in aberrant crypt foci, as well as the pro-inflammatory markers reduction, and also the decrease in the VEGF factor, as well as the increase in apoptotic factor. So this is a compiled study from all the flavonoid research in colon cancer. So when you take one specific compound, which is hesperidine, which is present in mostly oranges, not on grapefruit. So if you take this, hesperidine is mostly intact until it reaches the colon. And then when it goes to the lumen of the colon, it has a very significant effect on production of some of these short-chain fatty acids based on the gut. So that's a really advantage. While it is doing that, it is also doing a great help for killing some of these E. coli and other things, the same compound. This is, again, based on the human studies. So again, the reason there are a lot of contradictory results on hesperidine is because depending on how this alpha aminosidase activity in the gut microbiota, depending on the levels and how this is behaving with the gut microbiota. So if that is probably providing it, the hesperidine is one of the aglycon without the sugar molecule, which plays a significant role in cancer prevention. So again, in compilation of the hesperidine studies, it has its cardiovascular benefits along with glucose homeostasis, as well as blood pressure and some of the lipid profile. So the reason I'm showing this is one particular compound. Of course, there are several compounds in citrus can play a role. One compound has some significant impact on human microbiota also. So again, showing the epidemiological studies based on the literature. There are several thousand studies, 27,000 studies they picked from the literature. Only 12 studies were relevant for this meta-analysis. And that's the way the meta-analysis works. They take several and they have certain criteria to select, and then they take only small number of studies to understand the role of these study. So based on this, you can see that higher levels of the flavanol, quercetin, is one of the flavanol present in onion, which has significant help in impact on reducing colon cancer. Interestingly, the other flavanols, which are present in citrus on the bottom, flavanols, 3-ols, and anthocyanins didn't show that effect in this particular literature search. So now I'm coming back to pepper. So this is again a study growing in different locations in Texas, 3 locations. You can see the types of soil and the pH level. So what you see here is a significant reduction in capsaicin, which is the one which is pungent compound, which has good properties in cancer prevention, except it causes ulcers. But most of the beneficial effects are there in capsaicin. And you can see that the Uvalde location is significantly lower content than the other locations, even both in dihydrocapsaicin and capsaicin. Similarly, the quercetin present in pepper and the luteolin, it is again Uvalde location in quercetin and Weslaco location. They are kind of about 3 to 2, at least 100 to 150 miles apart, but the soil types are different also. So this is another project which are ongoing now. It's a large project with almost 26 scientists. It is funded by USDA with a $4.4 million grant. And basically, our approach is always a system-wide approach. That's why I was talking earlier, table to the farm. In fact, the USDA called me, what does table to farm mean? So I had to explain. That's a good thing because they're calling means we are probably getting some funding. So we thought it's a good way to convey that message. So it's basically a system-wide approach to address the crop management. When I say crop management, it includes soil, and we have soil scientists, agronomists, and also the outreach experts and the consumer experts and the safety, food safety. We are depending on University of Arizona for that. So what we did here is, it's again, the list of scientists involved in this from different universities. So the uniqueness of this is the same cantaloupes come to Texas A&M from all the 7 states. And we want to make sure that the same cantaloupe is used for a consumer panel, phytochemical analysis, sensory, except the food safety samples were sent to Arizona separately. It is a challenge for us to get it shipped and get it next day for 4 years. It's a 4-year study. So, but that's what we wanted to make sure that it's the same sample, so there is no variation in anything there. So the goal is, again, we did a sensory evaluation and also a consumer panel. And while we're doing it, we were interested in phytochemicals, right? So you can see on the top, the Texas, Georgia location has higher levels of beta carotene compared to Indiana. There is a difference in soil types. You can see those on the chart. And also previously, our scientists in Texas also compared the 2 varieties of melons. You can see the silty loam had higher levels of beta carotene compared to the sandy loam. That's the previous study, and the top one is the recent study. So there's a significant difference in the beta carotene. Same thing with, we also, as a part of this project, we have to develop some new varieties of melons, taking into consideration the food safety as well. So you can see that when we tested these, again, the beta carotene in Indiana is really very low compared to the other states in all the hybrids. So the other example is how is the composting makes a difference. Again, this is not our study. You can see that when you have the thermophilic bacteria, there is a significant changes in carrot with the flavonoids and carotenoids specifically. So what we did, of course, not because of that study, we did this study before. We developed this maroon carrot or purple carrot. In the beginning, when we took this to Texas Diabetes Association, people thought it is a chocolate. A lot of kids liked it because they thought it is a chocolate when we display that kind of setting. And it's also famous as an Aggie carrot, being a Texas A&M. So what we did is we did a lot of cell culture. We did grow in different locations and different types of soils. But what we found here is a human intervention trial. Again, this is a study in collaboration with the University of Arizona. You can see that when we gave these carrots to individuals with breast cancer, they were recovering from the breast cancer. They drank this juice for 3 weeks. So their beta carotene went up for 4.1.8 micrograms per ml. So interestingly, this is our study is about only $50,000 study, but there is a $20 million study that NIH funded. They showed very clearly if you have a higher levels of beta carotene, about 4.2 micromolar per grams, you can reduce significantly the reoccurrence of the breast cancer. So this beta carotene played a significant role in basically eating carrot itself will be helpful. And of course, our human intervention studies report is also there. So I'm going to touch on the last one, which I didn't think of covering earlier, but I thought 3 slides will make you a little bit more. I don't know how many of you heard this turmeric or how many of you eat this curry from India or anytime if you go to restaurants. So that has been a lot of attention in the United States. Now they spent about $600 million on research now on this curcumin, are basically coming from turmeric. So it is basically a strong anti-inflammatory component. So inflammation, of course, we get in by different source, smoking and all, but I highlighted in the red color how the soil, particularly the acid rain and heavy metals, impact inflammation here in this case. But of course, there are other several factors, which some of them we don't have any control. So if the inflammation is controlled, it's nice. But if you cannot, like in our system, a lot of the disease comes from inflammation, of course, and it's really chaos, right? So we have to control the inflammation at the right stage. So there is strong evidence. Again, there's probably 3,000 literature, which I'm not going to spend time on that, but at least if you look at on the left side, there's a turmeric literature, on the right side, the curcuminoids, which are present in turmeric. You can see that out of 1,000 articles on the turmeric and about 4,000 articles from the curcuminoids, 440 were selected, and the left side is about 140. They have clearly showed that these curcuminoids, as well as turmeric, has significant impact on cancer and human studies, not just only in animals in this case, and also cardiovascular and skin disease and inflammatory diseases. And the interesting part of this particular component, which compared to other phytochemicals we deal with, at the clinical level with the human studies, they tested about 8 grams per day. So turmeric has about 3% of the curcumin. So they tested that there's no safety issues. It's basically safer to consume until that level. So the reason I showed that is how is that soil impacts. So for example, there is a study done when you grow in a different types of soil, dark red to gray soil and red soil. Here on the left side, you can see that the curcumin powder is very high. And also here, the percentage of curcumin is very significantly high. Compared to the right side here, the red type of soil, you can see that the curcumin is low percent here on the right side here. So there's a big difference in how the soil plays on some of these bioactive compounds. So with that, I'm going to just display some of these recommendations. I think some of them are already discussed. I think I really applaud the presentation by Joe earlier about the education part. I think that's where we need to be really moving forward in terms of education. And also, just like human health, I think we have to consider soil as a healthcare. That's the only way to really understand problems and fix the problems. And as I mentioned earlier, system-wide approach is a key part to solve this soil and human health problems. Again, it's really hard to really bring in the team together, but I think it's important to really address this, all the experts, including the soil microbiome and human gut microbiome and the crop expert and soil expert to really address this issue. And again, as we already know about that One Health, and I think a previous speaker alluded that precision treatment, I think if you look at the top level, and the left side is basically the medicine and the agriculture. Everybody is really going towards the precision agriculture or precision medicine, right? So that's the key to really address this One Health issue. With that, I think this is again funded, some part of the project is funded by the USD NEPA. And also I'd like to thank the Texas A&M AgriLife Research for providing a lot of support, and Institute of Advancing Health and Nutrition, which is started in Texas A&M recently. And these are some of our scientists as part of the center. Again, I could not be speaking a lot of this data without their work and their some of these contributions to a lot of our work in the center. Again, we celebrated our 20th anniversary of the center and focusing on foods for health. And this is our group. As I mentioned, we deal with the key stakeholders all the time. We always have a biennial conference to discuss the issues about agriculture, including soil and human health. So with that, again, I'll leave it here. With thanking all my group and students and postdocs and scientists and some of the industry members.
Thank you very much. That was really interesting. We have some time for questions. You covered a lot of ground there, so there must be some here. Look at my hands here. Sam?
So I want to ask a naive question. So You mentioned about turmeric, and it's very, very interesting. And I'm from India, so there is people often talk about the importance of turmeric, of course, in diet. I was wondering if you found some difference in processed turmeric versus raw turmeric, like that you get from the field versus the processed that you find in the store.
That's a good question. We didn't do that study, but we did import some of these neglected non-turmeric product. Basically, they extracted these curcuminoids, the 3 curcuminoids, and then the 97% is kind of kept as a waste. We brought those. We isolated some of those compounds, not the curcuminoids. These are termurolines, and these are good compounds because they're not. You don't have to worry about the fat solubility, because some of these curcuminoids you need to have some oil or some milk with it, the curcuminoids. But the other compounds we isolated, you don't need to worry about it. They're basically absorbed very well. So, but we didn't really do those processing aspects because, again, we deal with the industry, we deal in Texas, what is their interest. So we don't really work a lot on curcuminoids in terms of research-wise. Yeah. It definitely, yeah, it's a great question. I think there is definitely a possibility. Again, I'm not going to say 100%. There is definitely a possibility that we may lose some of these compounds through processing, yes. But how much of the curcuminoids we lose, we don't know. Yeah.
Sarah?
Yeah, thanks for the presentation. Very interesting. When you were highlighting the various studies showing the presumed impact of soil differences on some of these phytochemicals, I saw some of the abiotic factors go by as well, like temperature between different sites and things. But did, for the most part, do those studies look at all abiotic stress factors and like precipitation regimes differences, as well as above ground biotic factors? Or what's your assessment of the state of the research when it comes to everything additional to soil when you're doing these site comparisons?
That's an excellent question, actually. So our study on grapefruit was not really done with the temperature particularly, but definitely we know that the temperature makes a big difference in lycopene synthesis, for example. If the temperature is more than 30 degrees Celsius, the synthesis of the lycopene is very low. And you need to have at least 12 degrees Celsius and above and below 30. So we didn't really do those particular studies. Our goal was, when we started this research, we wanted to really see why there is a difference in Florida and Texas fruits. We looked at lycopene content. That was for a marketing purpose. But interestingly, we found it's the same grapefruit grown on the same rootstock, except the type of soil are different. So we found that significant difference in lycopene content. So that's why Texas people always claim that their fruits are great, because they're not this lycopene, the taste also, the sugar levels also. But we didn't really look at specifically the different abiotic stress factors.
Lindsay?
I had kind of a related question. Thank you so much for the talk. It was really interesting. About the differences in the soil types that you were showing in a lot of those studies. Some of them you were, dividing up by location. And then the last one, you were dividing those soils primarily on the basis of soil color. I wondered what was really the key difference between those soils that may have contributed to some of those different plant compounds. Did you see any primary differences in things like organic matter, mineralogy, clay content, things like that, or nutrient content in the soil, other than things like texture and color alone? What was that?
That's an excellent question. The study we did on some of these vegetable crops, the difference we observed, at least particularly in onion and quercetin content, there is a difference of nitrogen. And the types of soil were a little bit same. But in terms of the curcumin, we didn't do that study. I just tried to show the health benefits and how that is probably a 30 years ago study I found in the literature, how the soil impacts the color of the turmeric powder as well as the curcuminoid content. There's not much study on soil impact. But the rest of the studies on carrot or other studies we did, we were really looking at growing location. And we're looking at, like the previous question, the temperature effect, how they really impact on phytochemicals. We really didn't look at other characteristics or other nutrients. So except in case of quercetin, we found that significant difference was because of the nitrogen content. And we know that having a higher nitrogen will reduce the vitamin C, for example. And of course, growers want higher nitrogen because they want to increase the yield. And then vitamin C is reduced significantly in any crop, for example. So there's no question about that.
Okay, let's take one final question. Ova?
Yeah, it's more of a reflection. Thank you for the presentation, first of all. I was intrigued by seeing the quercetin because it's come up in a couple of papers correlating positively with mycorrhizal colonization. So it seems like plants that exude more quercetin are also more highly colonized. So it's also maybe a signaling to initiate these beneficial associations in soil.
Mm-hmm. Yeah. Thank you. Yeah.
Okay, thank you very much. Appreciate this.
Thank you.
Okay, we're going to move to the next speaker. Oh, sorry. Covered a lot of ground there. Okay, our final presenter for today is Dr. Mario Ferruzzi. He is a professor in Arkansas Children's Endowed Chair in Digestive Disease and Nutrition Research. He serves as chief for the section of Developmental Nutrition in the Department of Pediatrics at the University of Arkansas for Medical Sciences. He also serves as the director of the Arkansas Children's Nutrition Center, a partnership between Arkansas Children's Research Institute and the USDA's Agricultural Research Service. He joined the Children's Nutrition Center as the director in 2021, having previously served as the David Murdoch Distinguished Professor at North Carolina State University's Plants for Human Health Institute and as a professor of food science and nutrition science at Purdue. His research interests are at the interface of agriculture, food, and nutrition science, and phytochemical bioavailability, metabolism, and impact to human health. Mario, we're looking forward to your presentation today. Thank you so much for coming.
Thank you very much. Just making sure you all can hear me well. Not where I thought I was going to be today. I was coming back from the American Society for Nutrition's meeting, and a lot of us didn't make it out of Boston yesterday. So somehow I'm in North Carolina, not in Arkansas. But I think what I'm going to do today is the task I was given was really to talk about the context of soil health and bioavailability of one class, focusing on one class of the bioactives that Bhimu and others have talked about today, and really is the plant phenolics. And in some ways, as we heard earlier, these are some of the category compounds that are perhaps part of Bruce Ames' plant-derived pesticides. But when I was first approached, I had to laugh a little bit because I thought they were reaching out to my brother, who's actually a PhD soil scientist for the USDA. So then it got to the point of bioavailability. I said, I think you guys had the wrong Ferruzzi there for a second. There's not many of us, but you had the wrong one. But anyhow, here are my disclosures, working with many federal agencies as well as the food industry. As you heard, I'm a food scientist as well as a nutrition scientist. And what I'm going to focus on is a class of compounds today. And this is kind of illustrative of many compounds, though. And I think Bhimu touched on carotenoids, touched a bit on phenolics in terms of the curcuminoids and flavonoids. What you see here are the diversity of what we see in plant-based foods, from simple phenolic acids, as you can see here, relative to the flavonoid compounds. And many of these compounds are present in monomeric or glycosylated forms in foods and beverages like cocoa and tea and red wine, but they're also present in complex polymeric forms that give characteristic astringent character to many of the foods that you see here, like cranberries with the type A procyanins, or dark chocolate, or really astringent red wines. And as we talk about bioavailability, it's important to understand this diversity, because these larger compounds, they themselves are not absorbed intact by the body. In fact, they interact they do interact with the soil and plant microbiome, but they definitely interact once we consume them with the human gut microbiome. So I had to smile when I saw Bruce Ames's level of exposure to plant pesticides, because it was pretty close to exactly where we're sitting now. This gives an example of the exposure or the intake that you could have from some common foods of these polyphenols. And you can see coffee is probably less known by many, but is probably the #1 source of phenolic acids in our diet, primarily as chlorogenic acid. So one One normal sized cup of coffee can give you about 665 milligrams. So if you're consuming 2 cups of coffee a day, you're getting over a gram exposure to these phenolics. And they are, of course, as Bhimu said, associated with many of the health benefits of fruit and vegetables in their diets, along with the carotenoids and others. But when I saw the NHANES number, I had to smile because here we are at about, with the NHANES data from 2016, 2020, the publication shows about 884 milligrams per 1,000 calories per day. So that gets you pretty close to that 1.5 to 2 grams a day exposure. So he was a smart guy. In many ways, I think you've heard a lot already from Bhimu about differences, agronomic factors, and even processing perhaps a bit on content. And all of these factors, plant genetics included, impact where these phenolics are in the plant, the food matrix. And what I'll show today a little bit is perhaps how digestible those plant matrices become, and to a certain extent, the level of bioavailability and metabolism that we see. Many of those factors, including soil, are certainly in the agronomic side and will have impact on that broader matrix, as I'll say in a second. What I'm not going to cover today is the other wrinkle to this, is whatever we discover is important in the field, remember the variation that you can get from the human side, depending on what our background diets are, gender, host genetics, nutritional status. There's lots of work on pathophysiological conditions, including obesity and diabetes, influencing our ability to metabolize, absorb, and metabolize these specific compounds. So I won't cover all that today. We're going to try to focus on a little bit more of what's in the other box and give you a little bit of background. So the Bhimu covered this, so I don't have to go into it too much. And I think we heard it as well in some of the earlier, referenced a little bit in the earlier talks, maybe not as much on the plant genetic side, but lots of agronomic factors are well known to impact levels of secondary metabolites, including phenolics, soil fertilization, cropping systems, pathogenic pressures. Some of this we know also because culturally it's important. In certain parts of the world, you will spray mold on and fungus on grapes to get certain to elicit more phenolic synthesis, or you'll pull leaves to get more sunlight into the grapes themselves, the light stress that they want to generate and elicit this response. You will not necessarily want to plant grapes in the best soil because you don't want a lot of nutrients, because you'll get more amino acids than phenolics. But anyhow, some of those have been reviewed already. I don't think we need to go into it, but some of it's purposeful in terms of eliciting phenolics for their importance in the quality of the foods. Well known that they can alter phenolic content and profiles. I would argue, though, what's not known is that direct connection between what happens in the field and what happens in the gut. And so there is a lot. I did a pretty exhaustive look. There's maybe a few papers that allude to it, but it doesn't mean that we don't know anything about this. What I'll show today is a lot of factors that we study from food that we know are affected by the agronomics, the plant genetics, the environmental factors, that we also know that they will affect bioavailability. It's just that the exact studies connecting those have not perhaps all been done. So let's talk about polyphenols or phenolics in general and how they're absorbed. And what you see here is the core of this is they come from plant foods or beverages. They have to be released through the normal digestion, starting in the mouth and then the stomach and through the small intestine. In the gut, you will see in the gut lumen chemistries that occur. So these polyphenols, PPs there, will perhaps oxidize based on the pHs that they are in the small intestine, exposure to reactive oxygen radicals. And then they go through processes of active uptake in the intestine. And these processes actually are mediated by ABC transporters, very similar to the transport systems and proteins that you see in plants, actually. Lots of folks that study auxin transport are also studying phenolics. They are metabolized by host metabolizing systems, primarily through conjugation systems, and those would be with the M. And then they are moved into the circulation and into the liver for distribution to tissues. Not any one tissue in particular has been observed to accumulate these, but it is rapidly excreted into urine. The bigger piece is perhaps what's happening for those that are unabsorbed, because this process is actually very inefficient, and the absorption occurs mostly in the lower gut and is primarily driven by phenolic microbial metabolites, as these are broken down. So upper gut, we hear the term bioaccessibility, just a term you should all understand. We hear that in the bioaccessibility of nutrients for plants. Well, we use the same concept for humans. So the fraction of bioactive released from the food or supplemental matrix by normal digestion and made available for absorption. And then later I'll say, and for utilization by the gut microbiota. But when we talk about bioavailability, we're really talking about the response in the body, and it's the fraction that's absorbed and made available to tissues for biological activity. So there's a subtle difference, but accessibility is used often as a predictor of bioavailability, and it's one that, depending on the models you use, can be a very good predictor of bioavailability. In the bottom there, you'll see as we get into the lower gut, microbial metabolism for many of the plant bioactives, including phenolics in a big way, are really required to generate the metabolites that we believe are actually eliciting the effects. Metabolism of these unabsorbed compounds yield smaller molecular weight, what we call postbiotics, if you will. There's lots of influences from food. So the source, the matrix, the type and extent of processing, Bhimu was getting into some of this. Part of that is driven by interactions between polyphenols and macronutrients, whether it's protein or carbohydrate or fiber, as well as micronutrients like calcium. Divalent minerals can disrupt lipid absorption. They can also be bound by phenolics. And then the overall profile of phenolics, they're phenolic-phenolic interactions. And this also carries down. If you have a highly fermentable fiber in your food, you're going to be changing the composition of the microbiota and perhaps changing the metabolism of the phenolics themselves. So with that in mind, kind of the flow of the way we like to think of how these phenolics from plants actually interact and deliver benefits is really this path by which they, in context of a food system, where they have all of those carbohydrates, fibers, proteins, lipids, other phytochemical classes, are reaching the microbiota, and what we would say is subtly causing shifts. I don't want to call them prebiotic because they're not in that traditional sense, but what they are definitely doing is influencing the microbiota, and they themselves also being transformed by them. But there are potential benefits both through the shaping of the microbiota itself, as well as through interaction and host metabolism, so our own body's metabolism, that deliver effects. But then the biologically active metabolites, whether they're phenolic metabolites, short-chain fatty acids, branched-chain amino acids that can be derived from those interactions, those postbiotics also deliver effects. So it's not the one compound that's in the plant. It's almost never that. It's most likely driven by what you actually see through the interactions of the microbiota. And this has been pretty well characterized, and with complexities of what you see in plants, whether it's simpler phenolic acids like chlorogenic acid in coffee or hesperidin from citrus, or the flavanthriols and procyanidins from tea and grapeseed and cocoa, you will see a series of microbial metabolism of these compounds, ester hydrolysis, C-ring fusion, fragmentation that occurs in the lower gut, leading to very, very predictable classes of microbial metabolites, benzaldehydes, hydroxyphenylpropionic acids, benzoic acids, hippuric acid, et cetera. And believe it or not, those of us in the food and nutrition space spent probably years looking back 30 years ago into the soil literature to understand how the soil microbiome was metabolizing phenolics. And that helped us identify, and I say us in the big field way, not us, our group, how these compounds are actually formed in the gut and then how they interact with human systems. But it is a complex array. This is a slide from my colleague, Katherine Kane. This is just happens to be one anthocyanin compound, cyanidin 3-glucoside, a traditional pigment in purple carrots and in many berries. And it will go through a round of metabolism to caffeic acid, dihydrocaffeic acid, all the way ultimately to hippuric acid or even phenol. And it is just like the London tube, because all of these pathways are interconnected, just like they would be in the soil. So it's ironic that what happens from the soil is also happening in the gut, and there has to be a link in terms of those metabolites, exposure, the bacteria, et cetera, that we see that's driving those. So just kind of to summarize, defining what we would traditionally say in the toxicology space is ADME, absorption, distribution, metabolism, excretion. For these bioactives, I want you to think first, though, as a food person, to think about liberation, right? So When I give you the example of grapes, one of the components of grapes beyond the anthocyanins are these complex flavanthriols, as I mentioned. These are the ones that provide the astringency. What you will see is if you consume grape seed extract or a red wine, you will absorb some of those compounds in the upper gut. They would be metabolized. In this case, you get a glucuronide metabolite that is in your blood. These are the series of classes you would now see the microbial metabolism, dihydrocaffeic acid being one of the most universally identified ones that is linked, for example, to neurocognitive, as well as to actually bone outcomes in clinical trials. What's interesting, as I mentioned, the vast majority of what's absorbed are those gut microbial metabolites. So if I was looking at traditional pharmacokinetics, you would primarily see that small area under the curve at the beginning, and people would miss the fact, and in fact, we did this for 20 years. We were focused on what was happening in the small intestine. We were ignoring that over a 48-hour period, you were seeing more like 30, 40, and 50% absorption, but they weren't the parent compounds. It was the gut microbial metabolites that are actually critical. So food matrix effects, metabolite profiles, and those microbial metabolite profiles, those are all key things here. And I just want to highlight some of the factors here in the last maybe 10 minutes to kind of get through this quite quickly. Food form and source can affect all of those steps. Formulation and food matrix, and you'll see here macro and micronutrient interactions, and then type and extent of processing. A lot of these have been heavily studied with mixes of models. A lot of in vitro models, which normally people stay away from because, oh, it's not a clinical trial. But when you're trying to get at mechanisms of food disintegration, some of these in vitro models are actually quite useful. There's several studies, some from our group, many from others, that really go into this. I put some pretty good references there, including some from our group, but definitely some from others, Torsten Bohn's group, for sure. And then I'll also give a couple of examples that I think are interesting because the plant genetic piece is quite interesting. But matrix versus no matrix. This was an easy study we were doing, actually not easy, but it's easy to explain in a sense that you compare a blueberry, but what happens if you take everything out of that blueberry but the polyphenols and you create an extract? So if you're going to talk about food matrix, the best way to test if food matrix matters is to take it all away, which is luckily what we had the opportunity to do when we were doing some toxicology work. And these are the breakdown products of two of the key classes, chlorogenic acids and the flavonoids in blueberry. And what we found is if you fed blueberry at 50 milligrams per kilogram body weights in these animals, these were Sprague Dolly rats, or you use extracts in a classic tox paradigm of low, medium, and high dosing, and you can see that. So if we match the same profiles, and this is a 90-day traditional toxicity study, you can see the berry phenolics, as we talked about, the microbial metabolites in the lower gut all the way to hippuric acid. And you can see that hippuric acid when you're feeding with fruit and all of that matrix, or even low doses of the extract, you see less of a response of those tail end metabolites over 90 days. You see more accumulation here. What happens is you get higher and higher on the extract without the matrix. You actually see less of the end stage metabolites and more accumulation in blood of the parent compounds and lower overall bioavailability. So what's happening there is you take the matrix away, you take away the ability to protect or shape the bacteria in a way that's conducive to making the metabolites. So changes will affect changes. And in fact, this was reflected in the bacterial diversities of those animals. When you got to the very highest level, you started losing the alpha and beta diversity of these in these animals. So again, not a perfect study, but it shows the extremes. And I just wanted to start with that one. We also think about a lot of chemical reactions that are occurring with these compounds. They're highly reactive. People think of them as antioxidants, but they don't really serve physiologically as antioxidants. They're just not that effective, but they are very effective at reacting with oxygen radicals in food and with other mechanisms. So this is just a summary here, and it's a little bit of an eye chart, but it gives you an idea that there are lots of reactions and susceptibility to processing: heat, light, oxygen exposure, moisture, pH, interactions with the macronutrients. This is a review that Howie DeBello written, my postdoc, a couple years back. when we were over COVID trying to find things to do. But what's actually really interesting about this is you can see the same kind of susceptibility through food processing. So it's not that this happens in a test tube, it happens at different stages of processing, and it is modified by the composition of the food. So whole grains, as they're ground, extruded, kneaded, doughed, and baked, depending on the end product, you will see different kinds of those interactions that affect the stability, but also the bioavailability. Green tea and black tea are the same botanical product. They're oxidized versus non. Fresh fruit through the washing, grinding, milling, etc. Thermal pasteurization, lots of different reactions. They occur at different stages. And this is there to illustrate the complexity of this question. It's really easy to say, I studied heating of a product and it changed this, but these are all the reactions that can be occurring. And this is not all of them. It's an example of the complexity. But it tells you that we're dealing with a very complex system trying to link just the food part, much less going back to the plant and the soil and even the plant genetics. But when you take the same exact grain and you process it a different way to try to simplify those comparisons, this is some examples from a study from Min Lee back in about 2015 on oats and oat phenolics and avenanthramides, phenolic amides, that are increasingly being studied. And what you see is you take the same oats and you wet cook them, or you jet puff them to make Cheerios. I don't want to give the company away, but you can read the paper, or Nature Valley bar, right? And it's the same exact starting material. It's processed differently. You see here the bioaccessibility, the release of that material in the upper GI is quite a bit higher for the processed product versus the wet cooked product. And why is that? Part of it is digestibility of those products, and we do change that. One of the mechanisms by which these are changed are interactions. These are some starch phenolic interactions that we were able to characterize in model systems, and have more recently been able to characterize actually in foods. Phenolics actually will bind with starch, form different complexes, and those complexes actually lead in some cases to resistant starch formation, depending on the nature of the starch, or loss of resistant starch and increase in rapidly digestible starch if the starch structure is different. So when you see high amylose versus amylopectin grains, and if what you're seeing is shifting that ratio, if it's the genetics or the conditions, you will see the interaction of phenolics with those starches change, is our hypothesis here. So that will alter that. Another example is phenolic interactions with minerals and protein. And again, if you think about this, phenolics, as you can see, this is a phenol side. They look a lot like what we call phenolic amino acids. Well, why? Well, because they're derived from phenolic amino acids biosynthetically in the plant, phenylalanine and tyrosine. So it's not shocking when we see that they can chemically, in this case, non-covalently interact through pi stacking. Hydrophobic amino acids and these phenolics will, in fact, be able to do those types of interactions. You can see here the calcium interactions that I mentioned earlier. It's a very good chelator. And so these interactions alter chemical stability, but also alter bioaccessibility and potentially bioavailability. This is a study that Kevin Croft's group in Perth, Australia, looking at addition of milk to black tea on vascular function. So tea is well known to modify blood pressure and flow-mediated dilation, primarily acutely through the absorption of flavanthriols like epicatechin, which then alter the vasodilation. They not alter it. They're basically altering the capacity for vasodilation. But then what happens over time is you see chronic effects in the vasculature that are driven by small molecule metabolites. However, if you put milk in the product, you reduce, theoretically, the bioavailability of the polyphenols. Well, actually, that's what Kevin did not see when they were looking at one particular metabolite, 4-O-methylgalate. It was responding almost the same. So he was thinking the bioavailability was the same. However, they didn't think of the broader microbial metabolites. So with some help from Colin Kay and his group when he was at NC State, we were able to compare those three groups for them and actually show that, hey, if you have milk in there, you're shifting the proportion of metabolites heavier from the propionic acid stage to the benzyl alcohol and aldehyde stage. So now what's happening is you're changing that proportion of what's in the gut, and that may be altering the responsiveness in the human. So at the end of the day, these types of interaction matter. And I don't say because we're really focusing on the soil microbiome to add milk, but if it's altering protein content of a vegetable or a grain, you might see differences in response to bioavailability. Again, it's illustrating the types of interactions you see. Big question in dietary guidelines for us is often comparing fruit forms. In this case, it's fruit juice versus whole grapes. And if you followed the debates in dietary guidelines over the last 15 years, and then what we hear in the American Academy of Pediatrics is fruit and juice are not the same product because you start taking fiber out. So again, if you start removing fiber, what happens? And so we used a very complicated in vitro model to get at every compartment, oral, small intestinal, to compare whole grapes and fruit juice. And we also did anaerobic fermentation. And just make a long story short, they're actually a lot more similar. On the left here, you see the total amount of bioaccessible phenolics. Concord and Niagara, we picked the two most common juice grapes compared to the whole fruit, matched from the same vineyard, everything. And what we actually found is, yes, there was a significant difference in bioaccessibility overall from fruit and juice for Concord, not for Niagara. But for the most part, the small intestinal release was highest for juice. Why? Because it's already digested in a way. Now, when you get to actually what crosses into the blood, this is cumulative transport, they're equivalent. There's no significant difference, maybe a little bit lower, but it was not significant for Niagara. But the big difference is what? They deliver in different places. So small intestinal delivery versus large intestinal, which means more colonic metabolites coming from fruit than from juice. So again, alteration of skin thickness, berry skin thickness, where you change the ratio of fiber to phenolic and everything else. Could these things matter? Maybe, but at the most part right now, we're trying to pin those associations from data that we only have data comparing these types of comparisons. We have data on the other side. We have to actually do the studies to be able to learn that. So where have we started crossing with the agronomic and what I would say biomedical? This is a review written by Connie Weaver. We were fortunate to be part of this overall program that was funded by NIH as part of it was actually part of NIC, National Center for Integrative Health, Complementary and Integrative Health. I always want to call it NCAM because that's when I started working with them. But what is the question at that point was trying to look at blueberries and bone health. And the reality was when they said blueberries, I said, well, what kind and where are you getting them from? Because that will matter in terms of if we have phenolic profiles, differences, and potentially even biological effects. So we started by looking at a collection of blueberry cultivars from NC State University. And this was 267 individual cultivars, primarily rabbit eye and high bush cultivars, mostly tetraploids, but I believe, and then in any case, what you see here is we did a PCA plot of just the anthocyanin profiles to show the diversity that's out there. And then made selections across that PCA plot to capture some of the diversity of the phenolics and, of course, everything else that perhaps is different based on the genetics or the environment. And we did a bioavailability study in rats, and I have a very stylized summary of this, and you can read Connie's paper for the detailed data. But we had three particular cultivars, Ira, Montgomery, and Onslow. I'll skip the SFH to B1. And then we had a low bush wide blueberry composite. And what you see here is the content of total anthocyanins in blue, and then the actual blood response with on the right is a yellow dot. And what you see is there was a difference between Ira, Montgomery, and Onslow. Montgomery was the lowest content, highest blood response. Why was that, right? So we began this process of trying to explore that. But why that became even more interesting to explore is that linked to the biological response that was observed here. Dr. Terry Bolito at UAMS had done the work with Ira and Onslow and Montgomery blueberries and the wild blueberry. And she found that only the Montgomery genotype was actually protective on bone in an OVX female mouse. And this was driven in part by endogenous antioxidant response, but then modulation of gut microbiome diversity. So it could have been more than just the phenolics, right? Could have been the phenolics in the context of that berry, which also drove the bioavailability. So this was where he got really interested in saying, well, how deep does this go? So if we have a blueberry and you think of all the factors that can be affected by the agronomic, by the soil, by the plant genetics, let's just call that plant genetics, because that was our focus. But in the context of this, it could also be the environmental factors. Where is it grown? How's it grown? All the soil factors. What's happening through processing? Did you consume it fresh? Did you juice it? Did you do anything else? And how did those things link to the absorption and the bioactivity? So this gets very complicated to unwind. And it's important to do it because we do want to see a G by E effects and perhaps even G by P or G by E by P. But a lot of this relates to the chemical composition first. And that's where a lot of the studies have gone. And my colleague at NC State, Massimo Iorizzo, has been doing a lot of this through his specialty crops center for the Vaccinium, VacCap specialty crop center, looking at the genetic pathways that are actually important for the type and form of anthocyanins. Why? Because these acylated forms, which are basically the anthocyanin glycosides that are tied to other phenolic acylating moieties like ferulic acid, caffeic acid, p-hydroxybenzoics. These are one step or two steps from amino acids converted by bacteria and or other enzymes. Are they responsible for altered bioavailability and physiological response? And so we've actually studied this. We did this as part of a project that was funded by the Foundation for Food Ag Research. We're in the middle still because we got slowed down. Obviously with COVID, we were in the key years of 2020 and 21, trying to catch up on some of that. But we actually looked, and I'll show you really quickly, if this is the paper that was published, some preliminary analysis on 66 accessions. And what we really were focusing on is how diverse is bioaccessibility of all of the flavonoids in this. And this gives you an idea of the variation of the percent bioaccessibility just on anthocyanins in this case. So you can see 2 crop years. I think the big thing to take out of this is if you look at this range, anywhere from the low teens all the way up to 85-90% bioaccessible for peonidin. Similar range for cyanidin. These are all different anthocyanin compounds, but you can have a very broad range of variation. And in fact, acylated derivatives were certainly much higher than non-acylated derivatives, making them more available for the bacteria and more available for absorption. Interestingly enough, the correlations when you look year to year on this were quite tight, but more importantly, there was not a clear correlation between raw content and relative bioaccessibility. So it wasn't just about what was in there, it was about how it was in there. And when you start plotting out absolute accessibility versus relative, we were able to identify, for example, some what we would call elite genotypes that we'd want to study more and be able to study them clinically. So when we want to do clinical trials, we don't have to do clinical trials on so many different berries. We can pick more to study. These are two different classes. So ones that you want that are really, really high in relative bioaccessibility, but a little bit lower in absolute. But the ones you really want are up here, high in absolute bioaccessibility, but also relative, which means they probably had lower amounts in there, but are outperforming what they should. Massimo's in the process now of studying the genetics of this to understand if bioaccessibility, bioavailability can become a trait that we can phenotype through high throughput assays, which is what we did here. And that can lead us to better breeding strategies and to better understand of what's happening there. In fact, it's somewhat encouraging that we're seeing moderate levels of broad sense heritability of bioaccessibility as a trait, at least in the preliminary analysis. I think when he sees he'll have a more clear answer, but it's somewhat interesting that we're there. So I'll stop with that because I think it's a lot, but I just wanted to kind of point out, linking back to the previous talks, in this case of phenolics or bioactives, they are definitely going to change in terms of the amount that's in the product based on lots of agronomic factors, including soil health. What we don't fully know is the extent to which they will be converted to metabolites. So that we know that the plant genetics and agronomic factors there, we do not fully know how much of these factors directly affect bioavailability and metabolism. But we know that if they can influence protein or lipid or fiber, then you will actually see effects, right? Main focus in this area has been the study of formulation, processing, and host genetics, as well as gut microbiome. So we have data on all the same chemistries that could be affected, all the same biochemistries that could be affected. They're just not yet applied in the field. So maybe an opportunity for some data mining and some machine learning to see what types of reactions can be predicted from interactions with soil microbes and soil health, as opposed to what we see in the body. Physical chemical interactions that impact release, stability, and the inability to interact with gut micro communities, those should be the targets of those studies, right? Which is, if we can compartmentalize what's happening, we probably can learn more about what's happening in the plant soil interaction to drive the overall plant quality from these products that then sets it up for the bioavailability piece. But again, I just want to stress, it's very difficult to find studies in that space that are doing literally from farm to that. A lot of people say they do that, and they do, but they're not necessarily studying those same factors that I would think your panel is studying there. So I'll stop with that and hopefully have time for questions if you need to.
Yes, thank you very much. Appreciate you doing that. And for now, what we're going to do is ask questions of all of our speakers. So if Joe's still with us, and Bhimu, and Katie, and Mario's already there, so you've got a chair. But if you would go up to the front, that would be great. And then we'll have two people on Zoom. Thanks, Joe, for staying with us. Okay, questions, folks. Lots to digest this afternoon. Okay, go for it.
Thank you so much for your presentations. And, this still leaves me with the question of how do we study human health impacts on that end of the spectrum? Because I think we've had such a great appreciation for all the complexity that's leading to changes in the content of these bioactives and their bioavailability and getting into the bloodstream. But then it comes to continuous health measures and getting that evidence, that seems to be a real bottleneck. So I'm just curious on thoughts there. So, for example, if we design a human intervention study and we don't have it focused on a specific disease, do we have something we can measure as far as reflecting physiological processes in the body that are working better as a result of these plant compounds?
I could take a stab at that. I think thinking of designing any one clinical study to address that broad area is, I'd like to say it's beyond my pay grade. That's a cop-out. I think it's probably not wise. I think before you get there, you probably can kind of, what I was trying to allude to is you might want to start with nutritional status markers first. Trying to get into the disease realm gets even more complicated by the nature of the study designs that probably need to be there and the burden. So for example, if you just were looking at absorption of critical micronutrients, or you were looking at changes in glycemic response of a product, or protein qualities out of that, I think those are things that we could probably do at different points in the chain and then string them together and do that. I just think that it gets really difficult to think about any one or even a series of 2 to 3 studies that are going to do this. I think you really want to focus on this area of quality of the product from the field, and then identify a couple of paradigms that are traditional in the food systems to put them through. But I'd be very careful about saying, oh, I want to take this into a trial with diabetics or with obese individuals and look at weight loss, or folks with cardiometabolic health issues and look at blood pressure endpoints and see if stage 1 hypertensives, the food is medicine folks are doing that. I think focusing on the product and quality side is probably where we can start.
Any other comments from you all?
Yeah, I agree with
Mike, need to turn the mic on?
Sorry. I agree with Mario. Only quick answer for that is I think whatever he said is probably I agree with everything. Is unless the National Institute of Health and USDA marry together, it's going to be hard because I have been pushing this about 15 years ago. I was here in DC, openly asked the question, how do you basically bring in NIH and USDA together to fund some of this research? It's not going to be happening. So that's the only solution, I think, in my opinion.
And even if they have, because they have had several RFPs together in the bioactive space, nutrition space, these are going to be one-off studies. You know, the interesting way might be looking more at population-based studies once you see agronomic systems and practice put into place, and you can look at diets that are derived from those foods versus others. So, you know, but those have to be perspective. Cross-sectional studies aren't going to cut it down the road. So, I mean, you have to.
We've got some more questions waiting. Oh, Anne, did you have something to?
As a follow-up, I mean, even if we move beyond disease markers to sort of that health promotion side, so the idea of eating better, even if it's not towards a disease target. I think these talks today really highlighted the complexity of difficulty in doing research in this area. So I really appreciate that. And I think the insights provided were really valuable.
Yeah. Okay, let's go to Kate, and then we'll go to Carla.
Okay, I hope this doesn't sound off the wall. Are guts like sewage treatment plants? And does retention time matter? And I think from a biodegradation perspective, right, with something moving through soil, you know, how long it stays there, where there are organisms to degrade it, will very much determine the outcome. So it's an issue of the transport, but also the rate of metabolism or degradation. And that's a function of population density. It's one thing it's a function of organisms who can carry out the metabolism. So is that an issue in gut also? Because I guess there's assumptions about how long it takes to pass it through. And any thoughts on that?
You're spot on. You're spot on. So it's come up in some of the talks today. There's no consistency also there's limited standardization in how we're doing a lot of these studies in nutrition right now on the gut microbiome. But most people focus on the lower gut, but we forget we have very metabolically active bacteria further up, number one. And then gut transit can be affected by so many factors, including your own physiology, but then the food itself. And you're 100% correct. You will get a different metabolite profile if you have something that's going through in a slower rate than if you do. But you'll also see different absorption up front. And so ironically, our guts are designed to disintegrate food. And so they exactly are like treatment plants of some kind. You know, I always joke everybody is scared of food processing, but the minute you eat it, you're insulting your body with that, and the body is processing the food far more than anything in a factory. But at the end of the day, you're spot on. All of those factors matter, and they're not always covered in large clinical studies because they become very complex.
Yeah, I just want to echo that. I can't agree with that more. And I think transit time is something that's often not thought of, and it's hugely important. It's why, sometimes in other world regions where children, are colonized with certain pathogens, and they're in these kind of chronic diarrheal states. Part of that is transit time, right? And it's also nutrient malabsorption, but that's a really important point you bring up. So fully agree what was said on that.
The only place they characterize it well is when they're doing a lot of work on glycemia. And they're looking at gastric emptying time is being monitored, but the rest of the GI may not.
Carlotta.
First, I wanted to thank you all so much for these amazing talks. You've given us so much to think about, specifically about all these complexities that we have to consider before nutrients are even absorbed by the human being. My question has to do with the variability in nutritional content of foods. Let's talk about fruits and vegetables and how that may translate into diet quality. And I'm wondering if you're aware of any conversions or attempts at conversions, even if they are theoretical, about how these different ranges of nutrient contents in fruits and vegetables could translate into something like a diet quality index, or you know, and making someone likely to fulfill the dietary guidelines. Maybe this question is for Dr. Patil and Ferruzzi, but
It's a great question, actually. So definitely there are several factors play a role on variability of this nutrient content, or particularly phytochemicals. So we have seen it over the time, and we thought first it's genetics, but it's easier to blame on genetics, but there are other factors, soil and temperature. So the best classic example is, I don't know how many of you heard the story about when you take a grapefruit juice, and if you're taking certain medications after 60s, you cannot take it. The reason being that there are certain compounds called furanocoumarins and flavonoids. The naringenin is one of them. Even though we have shown, at least even in the clinical settings, that if you take the grapefruit juice along with these medications, you can reduce the cholesterol, but you cannot prescribe to the consumers because there's a lot of variability of some of these components. I'm talking about furanocoumarins and naringenin in different grapefruits. So that's one of the reasons the drug companies still put a label on the grapefruit juice, on the drugs, that you cannot take this medicine along with the grapefruit juice because of the variability. So that's a complex issue in terms of solving it. I don't think we are there now to say, when you get a carrot, for example, in the market, you cannot prescribe, okay, how much of these nutrients you get it. I don't think we have a solution for that. There's a lot of variable factors play a role there.
Yeah, I think to build on that, Food Data Central, USDA's Food Data Central, the legacy database, they have a lot of different databases within it, is our national attempt to capture food composition. And that is actually in the National Program 107. And it's there in ARS, and it is in Beltsville, where they capture this. There are a lot of efforts underway now to improve that database in terms of not just updating data from 20 years ago, but to begin to capture the diversity of what consumers are purchasing. So that, in theory, would capture all of the factors downstream. May not be able to tease it all out, but you would get a better idea. So there are ongoing discussions, and I would certainly encourage you to reach out to the folks there. They're really great folks who are looking for, ideas and other workflows that can help them get there. It's a super heavy lift when you think about the number of foods that are in a supermarket, the 50,000 SKUs that you might see to get accurate data. But if they want to start like on fruits and vegetables, that's certainly there. There are data. The other place to look to start that is probably to work with the folks at the USDA and others that are holding the germplasm repositories. So those are really great opportunities to get more phenotypic data, including nutritional composition, bioactive composition, et cetera. So anytime I do a study on genetics, like we did it with the blueberries, that data is all transparent to them. They get all the phenolic profiles, all the vitamin C, organic acid profiles. When we do it with carrots, which we're doing right now, or sorghum, or whatever we're working on, spinach, where we're doing hundreds of different genotypes, we share all that data, we provide it. And I think more of that is happening now. A lot of other investigators, and as databases pull some of this in, I don't know if we can always make heads or tail of it, but you're going to see that mean value you see in the current databases, or what you pull off of, PubMed or dare I say Google, means are not that useful, to Bhimu's point, because the variation can be so immense. But there is an effort underway, and I think that's going to help us, but it is a little bit of a monumental lift.
I'd like to ask a question to Joe, and that has to do with, suppose you get one of these great discoveries, and you've talked about how hard it is to get the regulatory process through. Have any of your innovations really or discoveries gone on, and what's the next step? What would be your next step after this for these students?
Well, a great compound that has activity would probably start by being patented, if it was patentable, by WARF, our patenting and licensing entity, and then they would try to license it to a company. And it depends on how far we take it. And one of the things that's really changed in academic research that has an industry interface is that it used to be the industries would take very, very early discoveries. They would take a compound fresh out of the lab where we knew almost nothing about it. Now they want all the safety testing and efficacy done before they'll pick it up. So the question is how much I really want to do that work. And so one of the things, and I should have mentioned, because it is a conflict of interest of sorts, is I started a tiny company. I couldn't get the work funded. So I started a little tiny company and got the company to give my lab a grant to do the chemistry. It was the only way we could keep going. But that company now exists, and it could be the intermediary. It could be the industrial piece that does the pre-testing on all the things that the industry wants to see before licensing to big pharma.
So it would be one of the things one of the things that you would recommend is, and we may not necessarily use this, I'm just fishing here, is one of the things you might recommend that for bacteria, fungi, antibiotics, new antibiotics, that there is a pathway from USDA or somehow to bring more online?
Yeah, I think discovery is a really important part. Some companies have said that they screen 10,000 chemicals for every one that ends up on the market. I don't know if companies would agree on that, but that is one of the estimates. So discovery has to be ramped up, in my opinion, in order to meet the need. The other thing I think would really be relevant, this is kind of a different topic, but relevant to your study, is that I think soil erosion is very relevant, because one of the things that's always fascinated me is that vancomycin was only discovered in one soil sample. And with all the screening and rescreening across the world, most companies have, rediscovered some of the compounds, many of the compounds. But if vancomycin was only found in one soil sample, we don't know why that is, but it means that there are other sort of one-offs that could be extremely useful. And every time the soil erodes, we might be losing some of those. So I think there is a loose connection with soil erosion and your study.
Okay, threats. Okay, Sam has a question.
Yeah, I have a question about vancomycin and related to bioprospecting. I was wondering about this topic. So now we know that there are actually microbes that are resistant. There are bacteria that are even resistant to vancomycin, for example. And now from soil, they could get the compound. They could get this new compound called malacidines, right? So malacidine can even kill some of these vancomycin resistant bacteria. So my question is that we know that these antibiotic resistance and antibiotic resistance genes, this is part of the chemical warfare that microbes are involved in, right? And we can find antibiotic resistance genes in some of the caves that have been completely pristine and so on, right? So would you go back to the same, so in order to find compounds that can kill vancomycin resistant vancomycin resistant bacteria, would you go back to the same place where you found vancomycin, where you actually isolated vancomycin originally? You know what I mean? Like do you want to go back to the same place where you isolated vancomycin from, because you know that there are microbes that are actually that might
Yeah, the ecology doesn't exactly support that. There's very little evidence that these compounds are produced at sufficient levels in soil to act as warfare compounds and to kill the competition. People have loved that warfare model for decades and decades, that, you know, one organism kills the other, one develops resistance, all of that. And it's simply not feasible for most of the compounds. In most cases, to get production at a level where we can detect inhibition in the lab, we have to do something special. We have to ramp it up in some way. And then for industrial production, in almost every case, they've had to genetically manipulate the producer to produce more material or go to synthesis. So I don't think that we're necessarily going to find resistant or organisms that can overcome resistance with their compounds, because it's not clear that that would be an evolutionary advantage. But that said, if these compounds are signals, which is what I think most of them are, and there's starting to be some pretty strong evidence of that, then it could be possible that degrading that signal would have some sort of advantage. But I'm really speculating here. We really don't know the role of most antibiotics in the natural system. The only system where it's really, two systems where it's really been demonstrated evolutionarily is in the leafcutter ant symbiosis, where the ants carry antibiotic producing streptomyces on them to keep their fungal gardens clean of invaders. They're actually gardeners and farmers. They cultivate fungi for food. And the other one is in potatoes. The potato streptomyces community does seem to respond to antibiotic producers. And so you get more resistance if there's more of a producer there. Still doesn't prove that it's killing, but it's a little bit better supported.
Yeah. Oh, sorry.
I think this question is mostly for Katie, although I'm not sure. I'm curious about the breeding and breeding for increased microbial interactions, and because we know that historically we probably bred to reduce interactions with soil microbes. Is Bayer considering that, and what do you think the potential for that is? I mean, of course, the extreme being having a wheat plant fix nitrogen.
Sure. Yeah. So I think, certainly looking at that, and just to clarify, you mean breeding by sort of genetic modification? Yeah, certainly. Looking into ways in which those sorts of relationships can be enhanced. And there's a lot of gene editing sorts of experiments going on with that. How do you take, the certain features of certain microbes that are already naturally associated with that and try to enhance them? Yeah, definitely. But, one of the things I can say, even with gene editing, the regulatory paradigms still aren't clear. And so, so many things, I think, so much potential there for innovation and to work on that, and tons of work going on with that, and not just by us, but actually a lot of small companies, certainly a lot of university research, and I hope that's something that can remain on the table. But again, it comes back to that regulatory uncertainty. Kind of mentioned it before, but it's why we see, I think, so much consolidation in the agrochemical industry is because of trying to bring together resources to handle the regulatory component of it. I mean, it is true. I guess it's, it's true in pharma as well. There is a lot more sort of innovation that's being sourced externally for that very reason, to try to de-risk that situation. But yeah, I mean, there's so much cool stuff that could be done. And there is, I think, there's so much, I think it's been mentioned in past talks, there's so much, microbial dark matter out there that
the potential to learn new functions is tremendous. It's just, how do you actually take these innovations and get them to the market is really the big challenge. But I'm pretty positive about it. I think I have to be. I think we'll get there, but it's going to take a little bit of time, and in large part because for some of those microbial innovations, it's just not clear. Regulatory agencies aren't clear yet how they want to regulate them. I hope that answered your question.
Yeah, partly it does, but I'm also wondering about, as an ecologist, there's a reason wheat doesn't fix nitrogen.
Yeah,
right?
So
there's a cost involved in that.
Absolutely.
So just inserting a gene or multiple genes that allow it to do that is, so I'm just curious where you are with that. I know that people are.
I think that's just the story of agriculture. There's a benefit and there's a cost, and you're exactly right. We try to think about that in the sort of natural ecological context. Why doesn't that relationship exist? And sometimes to that question, I wonder if it's because many of the varieties that we have today. If you look at and almost every fruit and vegetable that we eat today, if you look at its ancestral state, it looks nothing like it is today. And so, I don't know that there honestly has been enough time for, were there some microbes that naturally were associated? I don't know, but there hasn't been enough time for those. We have modified things so quickly in, relative evolutionary timescales. I don't know if it's a function of, there was some deleterious impact of certain microbes being associated with certain plants, or if the opportunity just wasn't there in evolutionary timescales. I don't know the answer to that question, but that's one of the things I think about. And, we I don't know the answer, but I think that's one of the things we think about. But one of the things we absolutely have to do is field trials to kind of ascertain, large scale field trials to ensure that there are no significant deleterious impacts, especially when you're going down sort of the pathway of something that is like for biotech crops, for example. You have to ensure that it's nutritionally equivalent, that it's agronomically equivalent, because nobody will buy them if it's not. So I think field trials are a big part of that answer. And those are happening, I think, with greater frequency. So yeah, you're welcome.
Kelly?
Thank you all for your presentations. I echo what the other committee members said about how very interesting and inspiring they were. My question is also for Katie, but then anyone else may want to comment. I'm just curious in terms of management practices of soils and land that you've seen with agricultural production, are there any innovations that are particularly exciting to you in this soil health, human health interface?
Yeah, I think it's if anything exciting. I don't know that I could call out any one innovation. Of course, I'm biased towards microbes, so I think there's a lot of potential there. But I think it's using sort of integrated practices, whether it's from cover cropping, whether it's from low or no-till practices. I think integrating all of that, but integrating it at a sort of a precision level, because each farmer has so many different variables that they have to consider. I think if that's the thing that I'm most excited about. I mean, there's always so much interesting innovation too, again, from the breeding side. Again, not just, I think gene editing is really going to revolutionize what we can do in agriculture as well. So, I think from gene editing, which you also have a lot of excitement and enthusiasm for on the medicine side as well. So gene editing, precision, I think those are the two key things. But, I think it's really integration of multiple best practices, and really what is required is sort of good data metrics that can fit into these sort of precision systems, sort of big data, to give, farmers and the tools that they need to say, what do I need? What sort of management practice can I make on my, land? And also one other thing I'll throw out there is there's this whole new big space in carbon inputs and carbon farming. And that's a whole other topic for a whole other discussion. But can farmers, is there some reimbursement scheme where if there's some good metrics to say that they are, there's less outputs of greenhouse gases to the environment, can they get some reimbursement from that? But again, you have to have all the data and metrics to actually support that. So, yeah.
Thanks very much. We've got about a minute left for a question or a comment. And let's see, Kate's got one. Can you do it fast, Kate?
I'll try really hard. I'll talk really fast. Joe, this is for you. Soil structure, as I know, is vastly, vastly, vastly overlooked, despite how important it is, and regulating microbial community distribution, their interactions, their rates. And it's incredibly unsexy. I mean, people think it's hard to get people to appreciate the importance of it, like regulating water as well. And you may have mentioned something about microorganisms involved in creating soil structure. I don't know if you said it, but I wonder sort of where is that science? And obviously carbon is connected to it as well, and how to make it sexier. Read your book maybe about losing soil.
That would be one way. Yeah, it's one of my favorite topics because I think it's really sexy. I think it's incredibly cool the way microbes stick things together in their very special and thoughtful way that makes soil what it is. I mean, the look of soil is microbial. So I think we know that fungal proteins like lamellin are really important. I think bacterial polysaccharides are important, but it's still a pretty primitive science, I think. And so one of the things we were going to do, what I think I mentioned this in passing, is the engineer that develops these simple sensors is going to develop an electronic sensor that can hook up to a 96 well microtiter plate. And what you could do is put different microbes in the wells with some sand, and then see after a certain amount of time, it probably would take a while, the size of the clumps using his sensor, and be a very fast screen for many, many organisms. And it might be something that would be much quicker than the antibiotic discovery, but it would give us an incredible catalog of organisms that have an effect on soil. I mean, it's very reductionist. I recognize that, but I think it would be, and we don't have any reductionist work on soil aggregates. I think it would give us some clue about the frequency with which the ability to aggregate soil exists in the microbial world.
Thanks.
Thank you all very much. Oh, go ahead, Joe. You
didn't ask this, but I can't help but suggest, I hope your group is also looking at extremely intensive grazing regimes, because the highly intensive rotational grazing is said by its proponents to make beef carbon negative, because you get so much more plant growth that what's fixed is actually greater than what's lost. I'm not sure I completely believe those calculations, but it's certainly better, way better than feeding them corn. And of course, the grazed meat is much healthier for people. So I think and then it makes the soil much healthier because you get so much more carbon fixation, you get more root exudate and better soil. So I hope you're looking at that, because I think it's a really fascinating link between how we produce our food and human health and affecting soil health.
Great, thank you for that. Kara, did you have something? Okay, I want to thank all of you speakers, every one of you. That was just an excellent afternoon for all of us and brought up so many important points. And I also want to say that not only did he give us more food for thought, but I think we're going to have more discussions tomorrow on this report. I also want to thank everybody who's been online and stuck with us online for joining us. We do not have any future public webinar meetings or open meetings scheduled, but just keep in touch with our website because they may pop up. Thank Thank you again, everybody. This is really excellent.